To start our program, let's start our program with our university song. So I request all of you to please raise up at your positions. Thank you.
नित्यं हि विद्याचिन्तनम् नित्यं च विद्यामन्थनम् नित्यं हि विद्यासर्जनम् नित्यं च विद्याजीवनम्
Honorable dignitaries, respected delegates, respected teachers, and my dear friends, a very good and warm morning to everyone assembled here. I, Utkarsha Nimbarkar, feel privileged to extend my warm welcome to all present here for the global initiative of academic networks short-term course on basic immunology and immunotherapy coordinated by Department of Biotechnology, Sant Gadge Baba Amravati University, Amravati, Maharashtra. It is our privilege to welcome our honorable dignitaries by offering a bouquet. So I would like to request our respected Head of Department, Dr. Prasad Vadegavkar sir, to please welcome our guest with flowers. Coordinated by Department of Biotechnology, Sant Gadge Baba Amravati University, Amravati, Maharashtra. It is our privilege to welcome our honorable dignitaries by offering a bouquet. So I would like to request our respected Head of Department, Dr. Prasad Vadegavkar sir, So please welcome our host, Ms. Shama. We are eagerly waiting to inform that three different short-term courses of Amravati University have been approved by GIAN, and we are also very proud to share that this is the first ever program within Amravati University approved by GIAN. Looking at this beautiful gathering, I want to share one saying of a renowned French biologist Louis Pasteur. Science knows no country because knowledge belongs to humanity and is the torch which illuminates the world. This gathering is a proof that science do not have any boundaries, any religion, or it is not limited to only one field. Here, we only have a galaxy of curious minds. All respected delegates, on behalf of Department of Biotechnology, Amravati University, we are very thankful for your kind and auspicious presence as well as for your thumping response to this first ever course. We cannot thank you enough. As a student, I want to use this platform to request our Honorable Pro Vice Chancellor Dr. Rajesh Jaipurkar Sir and Honorable Dean of Science Faculty Dr. S.C. Raghuvanshi Sir to kindly include the credits of such glorious workshops in our choice-based KG system. It will be very beneficial for future. Thank you so much, sir. Now, may I request our beloved Head of Department, Dr. Prasad Vadegavkar sir, to give a brief about the GAN scheme as well as to introduce our esteemed faculty, Dr. Anshu Agarwal ma'am, to the gathering.
Very good morning. On the behalf of Department of Biotechnology as well as Santa Gadge Baba Amravati University, I welcome you all. Honorable Pro Vice Chancellor Dr. Jaipurkar sir, our Dean, Faculty of Science Professor Raghuvanshi sir, my friend Dr. Anshu Agarwal, all my colleagues and all participants. You all are here because of the GAN program, and you all are aware that Government of India has initiated Global Initiative Network Program for inviting the foreign faculty for the students of this country as well as the faculties and scientists. Earlier this program was only for the IITians. Since 2017, government has started sanctioning programs to the universities who have accredited A grade, and we enter into this scheme. I am pleased to inform you that we submitted 4 programs. Out of 4 programs, after reviewing, 3 programs have been sanctioned, and this is the first one. And credit goes to all our administration, including Pro Vice Chancellor, Vice Chancellor, Pro Vice Chancellor, Deans, who have helped at each and every step of this process. As you are aware that our university, with the help of him only, the accommodation has been made free to all, because we know that the students of this region as well as the students who are coming from other universities, all are from the, I will say, from the middle class, because they are coming from the places which are not at the metros. As I said that this program initially it was for the IITs, and now our universities have been included in this. We have tried to keep the registration fees as less as possible, and in future programs we will try to waive all the fees also. We will try to manage with the amount which we are receiving, or we will try to get some from somewhere else. This program is of a 5 days program equivalent to 1 credit course, and therefore the students have demanded that they should be included into their certificates. They have completed one additional credit course because this facility is available with the IITs, and we have to include this in our system, in the university system, because new program proposals have already been asked by the government of India for next session. So if you are running this type of courses, the benefit should be given to the students. So let me introduce the resource person for this course, Dr. Anshu Agrawal, who has done her PhD from CDRI Lucknow way back in 1994, and then she joined as a research scientist in um, ICGB, that is the International Center for Genetic Engineering and Biotechnology. Then she moved to France, Curie Institute for the postdoctoral work, and then she moved to Atlanta for Emory Vaccine Center. And from there she moved to University of California, Irvine as an assistant clinical professor, and now she is a full professor in health sciences. And her interested areas are human immunology, aging, dendritic cells, respiratory mucosal immunity, inflammation, vaccines, adjuvants, etc. She has published in all the top journals, which we consider the top journals, like a Nature, Journal of Immunology, Vaccine, you name the journal, including PNS. And she is, in brief, if I have to explain you, because we all are biotechnologists and we know what is the full form of NIH. Correct? So she is a reviewer of NIH, the project sanctioned by the NIH. NIH decides the policy for, you can say, the policy for all world in the health sciences. So she is with us for next 5 days, taking a course from basic immunology to immunotherapy. We will be end up in 2019 then, maybe from Pasture's time. In 5 days we will try to travel this complete journey. Sir, I would like to just inform you that this is a very heterogeneous group, because now we talk about there is no, as she has mentioned, there is no boundaries for knowledge. I will add, there is no boundaries for the science. So we are having the graduate students from the engineering background with the biotechnology. There are the graduates from the science. We have the doctors like Dr. Bote is here. Then veterinarians are also there. There are some people from the engineering background because they are interested in biomedical devices. So it is going to be a fun, because we are starting from the basic and we will see how to use the basic science and convert that into the applications. Our faculty, two of our faculty are also the participant for this course. And ma'am, I am also, I couldn't officially registered since I am the coordinating this, but I am the full-time participant for this course. So on the behalf of all of us, I welcome you. Thank you. Thank you very much.
Now, I would like to request our honorable chief guest of today's program and the Dean of Science Faculty, Dr. F.C. Raghuvanshi sir, to acknowledge us.
Chairman of today's function, Professor Jaipurkar sir, Pro Vice Chancellor, Sant Kalge Baba, Amravati University, Amravati. Resource person, Dr. Anjusha Agrawal Madam, Dr. P.A. Wadegaonkar Sir, Professor and Head Department of Biotechnology, faculty member of this department, and delegates. It's my immense pleasure to be with you for the inaugural function of this coursework. Dear friend, the performance of higher education institute in India is not very good. If we see the world ranking, in world ranking, our performance, I will not say bad, but our performance is not good. Why our performance is not good? It is analyzed by the central government, and it is found that our curriculum is not updated. As a academic head, it is my responsibility that curriculum should be updated time to time. The teaching method is not proper. There is some gap of learner, and we have to overcome this. And to overcome this, Government of India introduced this program. We are lucky that we got 3 programs by GIAN, and therefore I congratulate the head of the department, Dr. Wadegaonkar, and his team that they have given an opportunity to conduct such program which will be useful to our students. Dear friend, I am a Dean of Science faculty, but I belong to physical science. I don't know anything about the life science. Therefore, I will not talk about the immunology and immunotherapy. But basic is why this course is very important and why we should introduce in choice-based credit system. Definitely, from next year we will try to include this program in a choice-based credit system. This immunology is a science, is a branch of science. We deal with the immune system. If you study this, you know the functioning of immune system, and once you know the functioning of immune system, you can diagnose, you can give the reasoning, you can find the what are the causes of particular disease. And once you know the causes of the particular disease, we can treat the patient. This is a interdisciplinary topic. It is useful for medical science, pharmaceutical science, even it is useful for computer and engineering students. This and most useful for life sciences. Therefore, I wish my best wishes for this program. I thank to organizer for giving me this opportunity. Thank you. Thank you very much.
Thank you so much, sir, for your valuable enlightening. Now, I would like to request our respected chairman of today's program and pro vice chancellor of our university to share his thoughts with us.
Thank you. At the outset, I offer my sincere salutation to the motivational force of our university, the great social reformer, Karma Yogi Sant Gadge Baba, dignitaries on the dais for the inaugural function of the first GIAN short-term course on basic immunology and immunotherapy. Dr. Anshu G. Agrawal Madam from University of California, one of the greatest teacher, scientist and renowned teacher. My colleague in the campus, Dr. F.C. Raghuvanshi sir, Dean Faculty of Science and Technology, my colleague Dr. Prasadji Vadigaonkar, Professor and Head, Department of Biotechnology and coordinator of this short-term course, my colleagues in the university campus, teachers, and my dear participants from various states of India, ladies and gentlemen, very good morning to one and all. At the outset, I feel very proud to be here for this first international short-term GIAN course inauguration. The university is very happy to have this GIAN program. My dear friends, this global initiative of academic network, GIAN GIAN, was launched on 30th November 2015. And what I have been given information, this is the near about 500th program under the GIAN. Till now there are 486 programs done successfully under this international scheme. This is the international initiative by the Ministry of Human Resource Department, Government of India. And what Dr. F.C. Raghuvanshi has rightly pointed out, we are lacking behind in terms of higher education performance, and this is one of a kind of program to bring higher education internationally in the level of other universities. My dear friends, we have been sanctioned 3 short-term programs, and this is the first program. And this is a big opportunity for all Indian students, teachers, professionals to have such kind of program. Now, we all know that study, information, knowledge is now interdisciplinary kind of situation. We have linkages with other subjects. We have to take other subject while we perform research studies, and therefore but we have the variety of participants in this program also. There are near about 60 participants, right from doctors, biologists, botanists, and of course biotechnology people. And the participants are from Chhattisgarh, Kerala, Andhra Pradesh and Maharashtra. So Odisha too. So I welcome all the participants. What I have given information that till now there are 85 international professors working under the GIAN program, and one of the eminent professor, Dr. Anshuji Agrawal madam, is here with us. Madam, I welcome you on behalf of the university, and I feel very proud to welcome you. My dear friends, we must congratulate Dr. Prasadji Waregaonkar, who is the coordinator of this particular short-term program, and who is very keen to have this program in university campus, who was there having a great follow-up right from what I know from last 1.5 year. ये प्रोग्राम मिलना बहुत कठिन है और वाड़ेगांवकर साहब ने प्रोग्राम लाया है. So therefore I congratulate Dr. Prasadji Wadegaonkar sir. One demand is there that we must include the credit of this course in our curricula syllabus for the benefit of the students. So I assure you that we will go through all the technicalities, all the procedure, and we will definitely think positive. We have to go through the various authorities, but we will do it, no problem. My dear friends, I congratulate you and welcome all the participants, and I extend warm welcome to all the participants, and I extend warm wishes for the grand success of this short-term course. Thank you. Thank you very much.
Thank you so much, sir, for your valuable insight. Now, I would like to request Dr. Kove, sir, for vote of thanks.
It's my privilege to proposing the vote of thanks of this program. First of all, I am very thankful to Dr. Rajesh Jaipurji, Pro Vice Chancellor of our university. He is coming with the busy schedules and coming here for inaugural functions. Next, I am also thankful to Dr. F.C. Raghuvanshi sir, Dean Faculty Science and Technology. I am also thankful to Dr. Anshu Agrawal, so the professor in health sciences from California University, from coming madam here, and thank you very much. And last but not least, I am thankful to Dr. Prasad Wadegaonkar sir, the course coordinator of this GIAN course program. Sir already told me to conclude the vote of thanks in short. So with the permission of chairs, I here declare the inaugural program is over. Thank you. Here we can take the 5-minute breaks, and after 5 minutes, we will be start.
So first of all, thank you all for coming. And because it's an interdisciplinary course and we have people from all disciplines, anytime I'm going fast or if you have any questions, you can even stop me in the middle, it doesn't matter. And for some of you, it may be a little newer, newer subject. So as I said, because I've been dealing in this for so long that some things have become very easy for me, but you can always stop and ask me whatever is not clear, or if you want more elaborate explanation about any of the things I'm talking about. So we will first start with an overview of immunology just to see what is immunology in general. So this is a very basic thing which I'll tell you, and this I usually teach to people who have just entered college, this overview part, because this is more an introduction that, okay, later on if you want to do immunology, what it means. So what is immunity? We all, hear about that you should be immune against this and your immune system should be good. But what really is immunity? So it is the status or quality of being immune. And the word which, so it is basically if you are immune to something, that you are free from the possibility of acquiring that thing. So this has come from a very, in Roman Senate, or the people who were in the Roman Parliament, very very old times, they were they were not allowed to be persecuted for any crime. So they were there was a word known as immune there. So they were basically, they could do anything. They were immune to be, they could not be charged as criminal. So that's where the word immune has come from, and we use it as a, its resistance to infectious disease is one thing, but basically resistance to any kind of problems we encounter. Usually immunology started with infectious disease, so most of the terms have come from infectious disease. But now, as times have progressed, what we have realized is that we not only are susceptible to infectious disease, but also to pollutants and so many other things, chemicals. So we can no longer restrict it to just infectious disease. So who is credited with the discovery of immunity? The name is already mentioned, Louis Pasteur. So, he was a French, before Louis Pasteur, it was Edward Jenner. Yeah, and he's the one who realized that he's an English physician and naturalist, and we all no longer suffer from smallpox, but that was a major problem in the 1800s and 1900s, and even in the beginning of, so what he realized is that the milkmaids who were milking cows did not seem to get infected with smallpox. So that's when he realized that the cows seem to have these pustules which he thought that those pustules protect these milkmaids. So he just took a pustule and inoculated somebody with that pustule, which is really not possible nowadays. And the smallpox virus is vaccinia virus, and that's why the word vaccination comes from that. And this was the first example of vaccination, where we basically gave an organism and it protected the people from that particular disease. And it is a major achievement that by 1977 we no longer have smallpox in the world, because the vaccination has been so successful. I think this is the only disease, I think, against which the vaccination has been so successful that your generation doesn't require vaccination. Otherwise, so far we have not been able to master this in any other disease. So this is just to show what happened. So he inoculated a person from. What he found was, Jenner, that the milkmaids used to get the pustules, but they will never get the disease, just some minor form. So he took that. What he thought is somehow they are getting protected with this. So he inoculated another person with it. He also was protected. So from this, the science of immune came, that as you were putting something in these people, they were becoming immune to that disease. So this is how basically you can say immunology started. And it is so funny that it started backwards. So this is like the final stage of immunology where you get memory. And we started learning backwards, and now we are moving towards the beginning of immunology, how it is initiated. So, so this is very simple. So use of a vaccinia to prevent smallpox, so vaccinization and immunization also now known as. And basically the purpose of vaccine is so that you don't get that particular disease, or at least you are protected to an extent that you don't get the pathology of the disease. You may have some minor reactions, but not. So this is, as I said, these are very basic things. So as I mentioned, by 1977, the smallpox was considered officially eradicated, and there have been no incidents of it so far till 19. This is only in 1980, but for later years also, there's been no incidents. Only thing people consider is that, in fact, there's a lot of talk that there should not be even the virus being stockpiled at different places, because there's a chance of it being misused, or it can be reverted back. So, but that is still not taken care of. These are some other major immunologists who have contributed to various discoveries. So Louis Pasteur, I mentioned, so he was a French scientist, and he discovered the vaccines against cholera and rabies. And it is a very interesting incident. So I was in France, as mentioned. So they still have this Pasteur Institute there, and they have his lab preserved. So what he was working on was simply cholera toxin, and he used to grow this virus. But he went on, and this is, I think it was cholera toxin only, and it used to be in spinal cord of rabbits. So he basically went on a holiday, and his spinal cord was left on the bench, and the virus was supposed to be growing. So, but the conditions were not optimum. So when he came back, and when he took that virus culture, he realized that it was no longer causing the disease. So this led to one thing, that when you grow organisms in conditions which are not optimal for them, either the temperature or the medium, or basically you're changing it in such a way that sometimes they grow, but they lose their capacity to infect. So they will not cause disease. So it is known as attenuation. And that is one thing, that's also one way we try to make the organisms for vaccines. Many of the many of the vaccines we use, we basically grow the organisms in different, either the cell type is different or something, so that they grow, but they are no longer infectious or causing pathology. So all these, he realized that there are small organisms in the atmosphere or in different places which can cause disease. And this Ulrich basically realized that when we get a disease, something is made in our body, which is present in our blood, which can, if you give it to another person, it can transfer. So if a person has become resistant to a disease, if you give that serum to another person, the other person can become immune. So that's why he considered that there are factors in the serum which can transfer immunity. So these, Behring and Kitasato basically realized that these serum factors are basically antibodies. So the word antibodies has come, it simply means something which is made, it's anti-something. Anti means it's against, or it's opposite of something. And bodies is anything. It doesn't matter that it can be any entity. It can be an organism, it can be a chemical compound. So antibodies need not be only against an infection. It can be against anything, basically a chemical or a sugar or any possible thing. That's why we are able to react to so many different things and not restricted to infectious disease. And so as I said, the science of immunology started from the bottoms up. So most of the discoveries you would say that earlier ones were all based on antibody, because most of the work was also done in the blood and mostly on the serum. People were not thinking about cells so much at that time. So most of the work was focused on, protein which were isolated from serum, so antibodies. And by 1960s, first time we realized that there are cells known as lymphocytes in the blood, which are really playing this more important role in immunology. And the word lymphocytes, I'll come to, because the first lecture is very basic, so I'll come to it later. So what is the major purpose of the immune system? See, what you would like to do if you have a defense system in your body is that it should fight against the foreign things, but not fight against our own people, right? So same thing immune system does, that it fights against anything which recognizes as foreign, but it tries to protect. It does not protect the body from self, right? It does not. You don't want to destroy your own cells, and that, when it happens, causes major problems. You have heard of diseases like autoimmune diseases, lupus and all. In all these cases, this is what happens, that the immune system now suddenly starts to recognize our own proteins as foreign. And immune system assumes that anything which is foreign is harmful to us, and so we should try to fight it. So this is just to, it's a small cartoon just to show this, that self, nothing is happening here, it's trying to fight the fortress. So what are pathogens? Pathogens, all of you, since I think you are all biotechnology and most of you are higher students, so you already know the major classes of microorganisms are virus, bacteria and fungi. Parasites are protozoa and helminths. So these are older slides. You can add new things, but these are the major classes, you can say. And mostly other things are all subclasses of these things. So this is just to show you how much problem these small microorganisms can cause. So there was a major epidemic of influenza in this 1918 to 1919, and it killed about 22 million people in 18 months. And this was in Europe, and quite a bit of people were wiped off in this region. And malaria is, so many of the diseases, see malaria, TB, many of these diseases have existed probably longer than us, and we have still are in nowhere near finding any cure for any of these diseases. And a lot of work has been going on, but most of the vaccines also which have been developed have been more in the early era of immunology than later era. We are trying to improve those vaccines, but so far I don't think we have been very successful in many of these cases, except for a few, which we will come to. Like I think only the new vaccines I would say are vaccines against papilloma virus, which has come totally recently, which is new, I would say totally new. And I was saying that influenza, influenza vaccine, the problem is that every year you have to make a new strain. So a new vaccine. And there's a lot of problems are also associated with developing immunity with the influenza vaccine given in the previous year. It seems to be protective in some cases it seems to enhance response, which is not good for the people. So, but these microorganisms which cause so many of these problems, we are unable to see them because all these are very small things, right? So what are the beneficial consequences of a successful immune response is that you should be able to fight infections. So basically you should be able to fight infections, and for today's time, even cancers have become a major problem. Especially, I think it's not that the cancers were not a major problem in the earlier days, but because people are living longer, we see more of an incidence of it because there is a direct proportion, cancers and age are directly proportional. And simple reason is that if you think, it's a question of our bodies were made to function for 40 years. Basically, you're supposed to come here, reproduce, and then die. But now we want to live for another 40 years to now maybe 60 years. So the same system which is supposed to survive for 40 years is now surviving for another 60 years. So it accumulates a lot of changes and mutations. It's basically slowly decomposing or trying to kill itself. So that's why we have so many mutations and other things, and which leads to all the diseases which we see with the aging. And cancer is, one of the problems, because many of these mutations lead to cancer. Cancers, I have whole 2 lectures based on cancers and cancer immunotherapy, because of the cancers, the immunotherapy have become much more in use, because the newer therapies for cancer are no longer only chemical drug based or chemotherapy based. The new successful ones are more immunotherapy based. And the advantage is also that the immunotherapy is more of a universal kind of a therapy, which is not restricted to a particular kind of cancer. So that's why it is becoming very important. And those I will cover in the later lectures. Because that, I think, is a very good area which can combine most of the disciplines. And what are the problems which are associated with having an immune system is, one is allergy, because in all these cases, basically in all these cases, if you see, even though the diseases are different, the immune system is overreactive to things which we need, and that is why it causes these problems. Allergens are, like your pollen and other things. Slowly, somehow you become sensitive to them, and that's why, your immune system produces a special kind of antibody which is known as IgE, and that is what causes allergy. Transplant is simply that you're trying to replace somebody else's organ with somebody else's. So again, that's why we try to match, different antigens and things. But still, there's always some part which is a little foreign. So slowly, the immune system starts to fight, make an immune response against that foreign part, and that is what leads to your rejection of a transplant, because the immune response intensifies. So you will see that in many times in the beginning of the transplant, a lot of immunosuppressive drugs are given to prevent rejection of the transplant. And self, as you mentioned, is it leads to autoimmune diseases. An example is simply if you take lupus or rheumatoid arthritis, or even type 1 diabetes. In all these cases, so in lupus, what is happening is that each cell has a nucleus and it has your nucleic acids, but somehow our body starts to recognize the nuclear components, including the nucleic acids, from our own system as foreign. Our body is equipped to recognize nucleic acids from viruses and bacteria and other things as foreign, but somehow it starts to consider our own nucleic acids as foreign, and that's why in lupus a lot of antibodies and things develop against this. It leads to a number of problems. And type 1 diabetes is, it's different from the type 2, which happens later in life. Type 1 is usually in children and younger people. In this case, your immune system is thinking that your beta cells, which produce insulin, it considers them as foreign and tries to destroy them. In fact, it's not related to this, but one of the things where stem cell therapy is proving to be very successful is in areas where some of these cells have died completely, and especially in type 1 diabetes, it's showing a lot of promise. There's a clinical trial and some things going on in a university where they are putting these stem cells, and it seems to be able to make the beta cells regrow. So it is one of the areas which I think. So stem cells need not be always from the same patients, because one of the reasons things are rejected is because you have MHC antigens and other things, and stem cells do not express these antigens. So many a times people take it need not always be from the same patient is what I would say. Yeah. But right now, I would say the therapy is more people try to make from skin cells, iPS-derived cells, and then try to make stem cells. And initially people are not worried about, they are trying to take the same patient cells or trying to see if they can even grow beta cells. And then it's being put in the patients and see, if you take from the same person, there's never going to be any chance for rejection. So it's simply easier that way. Because so far, what we know is what we know. Like we think, okay, it doesn't express these antigens, but there's always something new which comes out, and then we realize that this is why it's not working. So what is the consequences of pathogens? Infection can lead to death, cancer, again, metastasis and death. So these are the harmful consequences. There are some evidence that a lot of infections also cause cancer. So as I said, these things will be covered in the cancer lecture, but it's simply this, that when infections occur, the immune system becomes activated. Sometimes it leads, many of the infections can mimic or activate some of the oncogenic elements in our system and leads to cancer. In fact, many of the viruses are associated with cancer. Example is the one I mentioned, papillomavirus. Papillomavirus is directly it's been shown that in most of the cervical cancers, there is a human papillomavirus infection which has led to this. So that is why the vaccine has been made against, the vaccine is known as Gardasil, which is for the human papillomavirus to reduce the risk of HPV infection and maybe in turn reduce the infection cause of cervical cancers. So this is just to summarize, but I think all of you are much more advanced. So simply the immune system's role is to protect the body from anything which is harmful, and beneficial consequences of the immune system or immune response is that you will recover from that infection instead of having other problems. And adverse consequences, we have already mentioned, the reaction to self-transplant and other things. So, this is just that if you are unable to control a pathogen, it can lead to different things. One is death. The other is that, it can lead to inflammation of your brain, meningitis, and a lot of different consequences can happen. But this is just one of a few small things which are mentioned here, that it can lead to cancer. It can lead to inflammation of different, inflammation of endocardium. This is basically for the heart and brain. I think the measles. So now we come to, so in all cases, you have like a small organ which takes care of different things. Like for digestive system, you have the alimentary canal. Then for neurological system, you have the brain and the spinal cord. So what is the immune system and what is its size? You know that, like, okay, this is particular kidney is this big and that is what is going to be involved in your neurological system. So immune system does not have a particular organ as such. And if you think about it, really it cannot have an organ because it's not restricted to one part of the body. Your infection or anything which problem can occur in any part of the body. That's why you have like small pockets of immune which are distributed all over the body. And these are known as lymph nodes, basically. All these blue things which you're seeing are lymph nodes. But the two major organs to a certain extent, you can say, of the immune system are spleen and the bone marrow. Bone marrow is the place where basically all the immune system, basically bone marrow is the place where stem cells are there, and all cells will originate from stem cells. So that's why the bone marrow is important, because immune cells also arise from the bone marrow stem cells. And this is just showing an architecture of the lymph node where you have, so the lymph node is an area where you will see more of the immune cells of the adaptive immune system, which we will talk about later. These are mostly lymphocytes. And the lymphocytes word has come because we have in our body two kinds of circulatory system. One where the blood flows, the other is where the lymph flows. And most of these lymphocytes travel via the blood also, but they seem to travel a lot by lymph. And that's where they were discovered. So that's why the lymphocyte word has come from. So how is the immune system organized? So there are two major arms of the immune system. So far, what we have been talking about, like adaptive, this making antibodies or lymphocytes, are all part of the adaptive immune system. And the first part of the immune system is known as the innate. So what is the major difference between the two? So innate is basically native or natural, or something which we have in our thing which does not change. So that is the first arm of the immune system, and it basically will respond to anything in the similar way. Though now we are discovering that there are a lot of changes in how they respond, but still there's a fixed response. Something will come in, it is going to produce this and try to contain the infection. While the adaptive immunity, as the name suggests, it adapts to the antigen or to the whatever comes in. It is also known as acquired immunity because people say that you acquire this immunity later. There are many words for it. Some people call it tailored response because it is like a person making a tailor making a cloth, or so a tailor making an immune response against that particular antigen. And this is the most important part of this adaptive immune response is that it is a learned response, like it can change. And this is what gives rise to the memory or vaccination. In case of vaccination, the reason you're protected is that your immune system, once it sees something, it learns that how to fight it better. So this is, if you think about it, any defense mechanism should have this particular property. Even your army, like it knows, okay, this kind of a threat has come before. So now you have already developed kind of defenses to fight that, because once something new happens, then you try to fight it, you try to gather things, but you are maybe not as efficient, but you learn. Next time around, you're going to be much more prepared for fighting it. Same thing immune system does. So the first part of any defense system is that it should try to contain whatever threat is there. And that is what the innate immune system does, that it is trying to stop it and trying to alert the adaptive immune system that you get ready. This is the kind of threat which has come. So this is how to gather resources. But I'm also trying to here contain the whatever threat has come, because you have to first do that to prevent it from coming in. So this, as we mentioned, what is the fundamental concept of adaptive immunity is that as you are exposed to pathogens, you develop memory and you are better protected against this. So this is like a very, whatever I have said so far, it can be organized in one slide. It's easier to remember in that case, is that you have different parts of the immune system. First is innate immunity, and that in the next part I will tell you that you have something, we have barriers in our body, right? One is barrier defense. So once something, a lot of it is taken care of by these barriers, that things do not enter a body. But then comes the cellular response. Once something penetrates those barriers, then our cellular response comes into play. And then these are all different responses which will happen. Chemical response, then we'll elaborate on all these, how these happen. And still we use many times the term cell-mediated immune response and humoral immune response. These are all parts of the adaptive immunity. But basically, humoral immune response is considered the antibody-mediated immune response, and here cells are involved. But both involve cells, really. So it's really not a very good, but these terms have been coined for a very, very long time. So in textbooks and in general, it's more easier for everyone to understand. So in this lecture, what I'm going to talk about is just simple, which are the cells which are involved in an immune response, and how are they slightly different from each other, and very basic knowledge, basically. So it's, for an immune response to be successful, it's not like only one type of cell has to react. It's a coordinated response of many different kind of cells which do their own functions, and that is all in total leads to the immune response. And it also depends on where they are located, that these cells will behave a little differently. And these are simply, as I mentioned, that in the bone marrow you have the stem cells which can differentiate into all kinds of cells, including immune cells. And this you have in the blood red blood cells and white blood cells. White blood cells are the immune, not immune cells, but considered mostly immune cells, and they develop from hematopoietic stem cells via hematopoiesis, and this is a highly regulated process. It's not that the stem cells will constantly make these immune cells. It is as and when required how much they should make. Everything is regulated. So that's why there's a concept of this niche, that only a certain amount of cells will be there. Like suppose you have 10 to 8 B cells in your body or B lymphocytes in your body. So your stem cells will not keep on making them. When some will die, then only the stem cells will make new. So the number is more or less, it's a repertoire which is more or less constant. So I think this I have already mentioned. The only thing which I will mention here is that from stem cells, there are two kinds of cells which can originate. One is the common myeloid progenitor cells, and one is the common lymphoid progenitor cells. It's simply that it's believed that there's two lineage differentiation from stem cells. One goes into lymphocytes, the other goes into making all kinds of other cells, which are all considered myeloid cells. Apart from the T lymphocytes and B lymphocytes, whatever cells are made, mostly considered as myeloid, originating from myeloid progenitors. And this is something I'm not going to go very much in detail. This is just to give you an example, and we will talk about these cells as we move. So all these are myeloid progenitor cells. So most of the cells, when we will talk, you will see that all these cells are part of the innate immune system, which are from myeloid origin. Except for now, you can talk about, which is not mentioned here, is innate lymphoid cells. But those are also from lymphoid origin, which have now become known in the last 6 to 7 years. And they are supposed to play a very important role, but I would still stick to these because they are kind of, in addition to these, they play a role, but they're not that. We are still learning about them. So I will focus on this for the time being. Then you have these lymphoid progenitors, and in this you have the two lymphocytes, basically T cell and the B cell. And T cells can be of two types, T helper cells and T cytotoxic. And as I will go further, I'll talk a little bit more. And this is B cells, and this is what I was mentioning, lymphoid cells. So these cells are also of lymphoid origin, but they play more of a role in the innate immune system than the adaptive immune system. That's why the name innate lymphoid cell is given. They are present in very, very small quantities in different organs. So cells in the human blood differ in number and lifespan. This is simply because some cells have a shorter half-life and they have a different kind of function where you need larger amounts of them. Some cells are very potent, so you need very small amounts of them. In fact, if they are increased in number, they can lead to disease. And that, as we go along, we will talk. This is just a table to show you that, red blood cells in our blood can survive for 120 days. Platelets. So neutrophils are cells. In fact, if you look at your WBC count in the blood, most of the cells are really neutrophils. And that's why you can see they can come up very fast and die very fast, because these are the kind of the first cells which will respond to any kind of infection. And if you look here, lymphocytes have an average lifespan which can go from days to years. And the simple reason is the memory which is generated is by the adaptive immune system, right? And those cells need to survive for a very, very long time. In fact, people have shown that even after 25 years, you have the plasma cell which was generated in response to a virus still living. So four main types of cells develop from the common myeloid progenitors. We will leave the red blood cells at this stage because these cells play a more important role in the immune response. So one is the monocytes, which basically makes macrophages when it goes into different tissues. granulocytes. In granulocytes, you have 2-3 different kind of cells, which I will come in the next slide. And megakaryocytes, these make the platelets basically. So the name granulocyte, as it suggests, is because these cells contain granules. Oh, sorry. These cells contain granules, and they are different kinds of granulocytes simply because they contain different kind of granules. So that's why the names have come. And so neutrophils are the first to arrive in any, kind of infection, and that's why they are in such high numbers and made, because their main function is to come, eat something, and go die basically. So they have granules which contain a number of enzymes which can digest the microorganisms. And the name, the neutrophil and this eosinophil and basophil, these are all different kind of granulocytes, has come because in the beginning the dye which was used to stain these cells, the granules, whichever dye was able to stain that granule, the name was given. Like in this case, the basic dye was able to stain the granule, so basophil was given. In this, eosin was able to stain, so that's how the name has come. So neutrophils are highly phagocytic. Phagocytic is basically, you can say, the eating function of these cells. Compared to neutrophils, eosinophils are not that phagocytic, and the eosinophils usually multiply in response to parasitic worms, but it also, if you see in many allergies, the number of eosinophils in the body increase and they cause allergies. They work along with mast cells and other cells. Sometimes you hear about eosinophilia. So that is what it is, simply that increase in the number of eosinophilic cells, which make it difficult for us to breathe. And basophils and mast cells also participate, are major components of, basophils act with mast cells to produce allergies because they produce histamines, which are, in fact, most of the inhalers and the drugs which are there against allergy are antihistamines, which will block this action. Yes. So these are different. So these are different components of the granules which are there. Like it has elastase and collagenase. Neutrophils also produce a number of antimicrobial peptides, like defensins, lysozyme, and so all these, basically will cause different kind of problems to the pathogen. Tissue remodeling, simply because, elastase and collagenase, because cells will need to move out if the infection is in the skin or wherever. From the bloodstream, these cells need to go there. So they need to produce factors which will allow them to migrate faster. In fact, these are some of the properties which are also used by cancer cells to migrate. So eosinophils again have produced this EPO, which induces formation of reactive oxygen species, which can kill worms and other things. In most cases, you will see vasodilation. Basically, whenever something is activated, it needs to move from one place to another. So you need your space or arteries to become expanded so that cells can move fast. So basophil and mast cell also produce this, a lot of these cytokines. See, when I say chemokines, cytokines, chemokines are simply, you can say, chemical mediators which are, and kines are anything produced by cells. So chemical mediators produced by cells, cytokines are proteins produced by cells, and these are just the ways cells communicate with each other. Because how are they going to talk to each other that, okay, there's some problem here, you have to come here. So these are different things which are secreted, and other cells have receptor for this, so they know what to do after this. If something is produced, the receptor will become activated, so it will tell. And also in chemokines, most of the time there's a gradient, that if a concentration of chemokine is in this area, cells will have the receptor, maybe because of an infection, the receptor becomes upregulated. So now it will move towards that particular gradient, that wherever the concentration is there. An example is, maybe I can talk about it later, but when you take dendritic cells, when they see an infection, they become activated. When they become activated, they upregulate a number of molecules, but they also upregulate a receptor known as CCR7, or so it's basically chemokine receptor 7. And because the major function of them is to take the antigen, whichever they are seeing, and then go to the lymph nodes to activate the adaptive immune system. In the lymph nodes, you have a very high secretion of this MIP3 beta. This is a chemokine which is there. I think it's CCL some 20, because now they have given new nomenclature. So that's how it knows. Once the receptor is up, it knows that it has to go there because there's a high concentration of something there which is attracting it towards. So it's a chemoattractant. So that's how the cells can know where to go. Similarly, cytokines also do this kind of function. They'll activate the other cells, and wherever the concentration is higher, you know that you have to move there. So these, the chemokines, cytokines, which are produced by cells, are very, very important in for our defense system. In fact, these are very important for our diagnosis also, because these are mostly produced in response to some kind of a problem. And in fact, there are different kind of cytokines which are produced in response. Okay, I think I've gone back. Okay. So we talked about granulocytes. The other major cell type which is involved is the monocytes. And so one of the major properties of the granulocytes, which I should have mentioned, is, just go back a little. So if you see in all these cases, the nucleus is multi-lobed. It has like, it looks as if it has like 4 or 5 nucleus for that matter. And that's why these are also known as polymorphonuclear cells, that they have polynucleus, like multiple nucleuses kind of thing. It's mostly lobed. It's one nucleus, but it has more lobes. So, and that is how you, in fact, you can recognize, yeah. Lobe structure, I think in this case, one of the reasons which I think is that these cells have to respond very fast. So they have to make a lot of proteins very, very fast. So the nuclear machinery is, I would say, required in much larger quantities, and they die very fast. Otherwise, no particular other reason has come up as such. And okay. The other is the mononucleus. The other cells are all mononuclear. You will see that the nucleus looks like one small round. It's here it's showing lobe, but it's mostly looking, it is a more round cell kind of a thing. It doesn't have those different lobes which are there. And I think this nomenclature has come because mostly when immunologists work, they need to separate the red blood cells because they interfere with most of the things. So we use a density gradient centrifugation. And the density for cells which are having multi-lobed, because they are more dense because of the granules and the multi-lobe, the density is different. While the cells which have just one nucleus and no granules is very different. So it's one way of separating them. So you take the density, when you use FICOL, basically you separate the cells which are mononuclear. And the other problem is most of the studies are also done on mononuclear cells because the neutrophils and eosinophils are very hard to culture. In fact, the moment you isolate them, they start to die. So that's why it's harder to study them. So yeah, the moment. So these monocytes are, as I said, when they go into tissues, they become, instead of rounder, they become more bigger cells. And they kind of adapt to wherever they are going. So just to give you an example, when monocytes go into the brain, it's believed that the microglia, they become microglia. In case of liver, they become the Kupffer cells. So wherever they go, they become a little different, and their functions more or less remain the same, but they do acquire organ-specific function. In fact, many of this, in the fat tissue, most of these macrophages assume a different kind of morphology known as foam cells, which accumulate a lot of lipid droplets in them. So they change their morphology in that way. And then you have these dendritic cells, and the reason, so there are two kinds of dendritic cells in the body which people become confused. One is that when you talk about dendritic cells, even the brain cells are known as dendritic cells because they have these spine-like structure. And the immune system dendritic cells were named dendritic cells because they also have these spine-like structure. And the dendritic cells in the body are much less because, see, the granulocytes which we were talking about, they try to defend, but they do nothing. They will secrete these cytokines, chemokines, and things, but they don't really alert the adaptive immune system. They will not. They will alert, but they don't do anything direct contact with the adaptive immune system. While these monocytes and dendritic cells, they basically act as the bridge between the innate system and the adaptive immune system. The way it is that these two are considered antigen presenting cells. What they will do is they will react to whatever pathogen has come. Similar to neutrophils and other things, they will also phagocytose, but they will not die. They will take that antigen and take to T and B cells to make them, basically T cells to make them activated. That's why they're known as bridge cells, which are both part of the innate immune system and also are important for initiating the adaptive immune response. So because of this property, these cells are most cells are distributed all over the body, but dendritic cells are also known as sentinels of the immune response, and they are present in just below your lining of the skin, lining of the mucosa, nasal mucosa. Because if you see, if something has to come in, it has to penetrate this barrier, right? Either nasal, intestinal, or skin. So these cells are present just below, so that they can take that whatever is coming in and now go to the adaptive immune system to alert them. So that's why they are distributed in all over the body at the points of entry of pathogen. And the number in the blood is very, very small. In fact, it is like 0.1 to 1% at the most. So it's harder to study them in the blood because blood, they're usually there because they're being made in the bone marrow. They're going to the place where it is required. So that's why some percentage is there in the blood. But mostly they are more in the tissues distributed. And the dendritic morphology comes, so in the beginning, if you look, these cells also look more or less round. But when they become activated, they have these spine-like projections. And the spine-like projections are not just for fun. They have an important role because if you have these projections, the surface area of your cell is increasing many-fold, right? So it's allowing many more, and these cells are much bigger than lymphocytes. So one cell can bind to at least 20 or 30 more lymphocytes. So that way you need one cell which can activate maybe 50 lymphocytes. So you need much less number, but they can do a number of, they can be functional to a large extent. Size varies a lot because the smaller one is similar to monocytes, about I think 5 to 10 micron. But when it becomes bigger, it can, because of the projections, it can be pretty big. There's no fixed number because I've seen that people say that sometimes they see very big, sometimes they see very small. There are different stages of differentiation. Could be. I'm not 100% sure because I have not looked at the size that carefully. So amongst the cells we have mentioned, these two cells are considered to be highly more specialized for phagocytosis. But the difference between these two is that macrophages can act as antigen presenting cells, so they can present antigens to T cells. But it's the immature dendritic cells which I mentioned. So immature dendritic cell is a term which is basically, it's newer in the sense unactivated dendritic cells is people call them immature. And mature is when they become activated. So I think I will have slides later, but it's simply this. if you see, in the beginning, what the dendritic cell is supposed to do, that it's supposed to see a foreign antigen and capture it. So the phagocytosis function is very important at that time. It doesn't need to do anything else. It's supposed to recognize that particular pathogen, take it in, and now digest it to present a small portion to the T cells. So initially, when it is immature, it is highly phagocytic to take up the antigen. And once it takes up an antigen, now it needs to present that antigen to T cells. Then it upregulates, it becomes activated or mature, as they say. And at that time, it upregulates a number of proteins like MHC, co-stimulatory molecules, and other things which are required now for that function. So that's why the morphology of the this dendritic cell is very different before and after. Immature and mature, the type of proteins which are expressed are also very different because the functions are very different. In one case, it is phagocytosing. In the other case, it is presenting. So that's how people, and it's an important thing to, it's very important to identify which state the dendritic cell is in, because it will tell you if it's making an immune response against that particular thing or not. So it's used in diagnosis in many types of, to see how the immune response is functioning in many types of cancer and against infection. So that's why the terms immature and mature have come, because there are certain molecules which will be upregulated, which you can see. And if you see, I mentioned that both macrophages and dendritic cells can present antigens. But the reason dendritic cells are considered more important is it's believed that the initial response, the first response or initiation, the first recognition is by dendritic cells, and then the macrophage number is more. Then they kind of carrying forward is more by macrophages and B cells. But to start an immune response, it is more dendritic cells which are important. So there are three main types of cells which can develop from the lymphoid progenitor, the B lymphocytes, the T lymphocytes, and in T lymphocytes there is a small subset which is more like natural killer cells, and they are also known as NKT cells, and these are natural killer cells. So the name B lymphocytes has come because it was discovered, these B cells were discovered in the burst, in the chicken, in the tail, the bursa of the chicken, that's where they were discovered first. That's why the name B came from. But also because now they're made in bone marrow, people say that they should be known as lymphocytes, but all cells are made in bone marrow. So, but the real reason the B name came from is from there. And the T lymphocytes has come, they're also made in the bone marrow, but they undergo a process of selection in the thymus. So that's why they're known as T lymphocytes, from the thymus. Yeah.
Play an important role in antigen presenting cells. Is B lymphocytes is less? Will they play less role than dendritic cells?
No. So B lymphocytes can also present antigens. It's because three cells are antigen presenting cells:
Yes,
the dendritic cells, macrophages, and B cells. Yes. But the initial first part, the initiation of the antigen presentation is usually by dendritic cells. B cells a little later, because the B cells are not usually present in the areas of entry of the pathogen. So they are present in the lymph nodes. So once the antigen reaches the lymph node, the B cells become more active. So it's a little later. Like if you see, in the first one day, it's going to be more dendritic cells. Then later on, it's going to be more B cells and macrophages which will take over. It's not that they cannot present antigens. But T cells cannot present antigens. And one of the problems with T cells is they cannot see any antigen. They can only see antigen when some other cell is showing them the antigen in the context of MFC. And that's why we need antigen presentation. Otherwise, we may not need. The NK cells, on the other hand, are very important cells. And the name, if you see, is natural killer cells. So their major function in the body is to survey any cell, and it doesn't matter if it is our own cell. If it is not looking normal, if it's expressing some protein which is abnormal, especially in case of cancer cells, these play a major role because cells become a little transformed. So anything which doesn't look normal to them, they'll kill it. And we also have in the T cells another subset known as cytotoxic T lymphocytes. They also do the same function. But the difference between natural killer cells and T cells, cytotoxic T cells is, those will only kill cells, antigen specific cells, which we will, like, they cannot kill indiscriminate, they cannot kill anything. NK cell in general do not need help from anything. If they find anything abnormal, they will kill. So an example is when a virus infects the cell, it will change the proteins which are expressed on the cell. If NK cell sees some different protein, it will kill. And it also kills via granules, but the granules which it secretes are different from what you find in neutrophils. In this case, it secretes basically this granzyme B or granzyme A, granzyme, and it also makes a protein known as perforin. So the perforin, what it does is it creates pores in this other cell membrane, and then the granzyme goes in and it kills the cell. So it has like both the things which it requires, so that whatever it's making should be able to enter the other cell. So it makes a protein known as perforin, which basically perforates the cells in a way, the membrane, and then the granzyme can go in and kill. And the lymphocytes, they look, if you look under the microscope, they look small round cells. They are usually much smaller than the innate cells, because as I mentioned, one dendritic cell can bind 50 or more, or 20 definitely. So size has to be different. But the way you can differentiate them is because on their surface, the proteins which they express are different. And that is, these proteins are now known as clusters of differentiation, of CD number, which you will see a lot of times, like CD4, CD8. So these are basically just batches of proteins which are present in high density on that particular cell type. So these are just some of the CD molecules which I have mentioned here. So CD3 is, all T lymphocytes will express this particular. So if you want to see in your thing if the lymphocytes are T cells, then you can take an antibody and stain it with CD3, stain the cells with CD3, only the T cells will be marked. So, and because why it is expressed on such a high amount on these cells, because it forms an important part of the T cell receptor signal transduction mechanism, which we will come again later, as I said. So the T cells we divided into two different kind of T cells. One is CD4 T cells, and these are important because these are molecules which will bind to the MHC2, so they are important in antigen presentation. So, and CD4 T cells are usually considered T helper cells, and the name has come because their main function is to help B cells produce antibodies. Their main function is mostly to provide help to other components of the immune system. That's why the name helper cell has come for CD4. And the other component, the CD8, is basically the other type of T lymphocytes, which is the cytotoxic T lymphocytes, which will also kill. It's also very important for viruses and other things. It kills in the same way as natural killer cells, but only antigen-specific cells. So CD5 is also expressed on many lymphocytes. I would say in mouse, it is used to distinguish between two different kind of B cells. But for right now, I would say just focus on CD3, CD4, CD8. CD16 is, so there are many different kind of CDs. Right now, I think the CD number has gone to about 400. So it's no point trying to learn what all of them do and what is their function is. Whatever you require, just worry about that much. And there's a big chart which comes, put it there and just see each time somebody tells you what the CD is supposed to do. But this is important. This is what I'm going to mention simply, that this is all B cells will express CD19. So one of the advantages of these CDs is that some are present on many cells, but some are unique, like CD3, which I mentioned, will be present only on T cells, or at least at a much higher density on T cells. Now people are realizing, okay, a little bit is expressed here and a little bit is expressed there, but mostly it will be present. Similarly, all B cells will always express CD19. So if there's any reason you want to get rid of B cells, you should aim for CD19 specific antibody or something like that. So this is highly important because in your blood, if you're trying to look at different kind of cells, this knowledge of which cell expresses which CD at a high concentration is how you distinguish between different cells. And that is what I'm going to talk about. After my adaptive immune lecture, I have a whole lecture on flow cytometry and how you identify these things, because it involves microfluidics and which is important for engineering students also. But this is just to say that there are many molecules which are expressed on different kind of cells. So sometimes you need to use not only one marker, but a combination of 3 to 4 markers, that it should not express this, it should express this, it should express this to an intermediate level to identify a particular cell type. And these CDs, apart from identification, also have functions. It's not that they don't have any functions, because many of them are involved in their cell receptors. So one of their thing is to transfer information from the top of the cell to the nucleus. So the cell can make the proteins and things which are required, because these receptors will get activated in response to something or the other, right? Once they are activated, that means that the cell now needs to respond to that particular thing which has come in. So signal which is transmitted from the receptor will go to the nucleus. Whatever kind of proteins or whatever kind of things are required, now the cell will synthesize that. So most of these CDs also have that function. Like the CD21, this is the B receptor requires these 3 components for it to function. CD28 is a co-stimulatory molecule which is also required by T cells to become activated, which we will, as I said, come later. And these are some molecules which we use for distinguishing, like NK cell. Neutrophils express CD56, I think. But it's like all these are used for different things. We will just go with that particular thing. And this I already mentioned. So the natural killer cells are not phagocytic, but they have this property of surveillance. And let's see if the animation will work, then we can. Okay, we'll show it some other time. But it just shows that how it kills a particular cell type in this movie. Maybe we can, after this, we can ask them to connect and then we can see. So basically, they constantly survey our body and they look for infected cells, cancerous cells, and basically they just, if there's any cell surface change, they will kill that particular cell type.
How many NK cells will be there in the young people, and how many NK cells in the old people? Because cancer is coming mostly at the age.
So mostly the number in all these cases, the cell number is, there's a range of cells because our humans population is highly heterogeneous. So these numbers vary. So for each population, usually these pathology labs, they do a number of survey, like say 5,000 young people. Then they'll find that in all these people, there's no disease, but the range is from say 10 to 4 cells to 10 to 5 cells. So that kind of a range is established, which varies.
But it is declining, right? When we
grow old. So in many of these lymphocytes do not really decline with age, even though they are not newly made, but many of them stay there for a long time and they become dysfunctional. So the numbers may be constant, but they are not functioning,
not efficiently
working. So not always the number is a very good measure of saying that okay, these cells have disappeared completely. Both things are important. You have to look at the numbers first. If there's no change in the numbers, you cannot assume that it will function. I'll give you an example with age, because what has happened is over the years, all of us are infected with CMV virus, cytomegalovirus. It doesn't cause any disease as such in most people, but there is constant reactivation because it's there in the atmosphere. So the T cells are becoming constantly reactivated. So as I mentioned, we have a repertoire. Suppose we have 10 to 8 T cells. Because of this constant repertoire, now instead of the 10 to 8, which are all different specificities, we have only 10 to 6, which are different specificities, and the rest 2000, the rest 10 to 2 portion is now only devoted to CMV. So your number of cells is the same, but most of them are now responding to only one kind of viruses. So you have less number of cells which can respond to other things. So with age, this is one of the problems which we see, that it seems to have a lot of accumulated cells over the years, which are for different things. But now you don't have new cells to fight for new infections or new things which are happening.
So the stem cells are exhausted.
So stem cells, so that is also kind of debatable because stem cells, people have shown, there was an experiment which was done in Berkeley where they have basically connected a young mice and a old mice. So what they saw was that the stem cells from the aged people, when they go into young microenvironment, they become functional. So it's the microenvironment or there are factors in the blood or factors in the tissues of the aged people, which is making the stem cells to not work very well. So if that atmosphere is changed, they are able to function. So we cannot say that the stem cell is exhausted. It seems they are encountering an environment which is making them non-functional. So there is also niche is not working. So that is one thing, but it's not that they are always very functional, but they seem to function. At least, let's say, if not to 100% efficiency, to an 80% or 70% efficiency when you give them the right microenvironment. So that's why people are, there is an initiative to identify factors in young, they basically call it regeneration factors in young blood, which can basically activate the immune system of the old blood, something which can be given, maybe some protein or something which can be identified, which can enhance the function of stem cells and others. Because it's always easier to activate something within the body than to take it out and put it back.
Ma'am, you are saying something on the first or second slide. So if you give a serum is having antibodies. If you give to the patient who is having disease, so it can go and minimize the pathogen level. So this is just a guess. Okay? So we can take a blood from everybody is carrying TB here. So we can take a person blood from TB resistant patient to TB sensitive patient. If you transfer the serum, will it work?
To a certain extent, it should work. It depends how much protection it is going to offer, what kind of a problem the other person has. It can neutralize if the bacteria is there in the blood, right? The antibody, that's what it's going to do.
Cell, it will not act.
It will not act because it will go and bind to the TB bacteria which is circulating and get rid of it, but it will not be able to penetrate the cell. So that's why this is like a passive therapy, this serum transfer. So it works only in, it's more, nowadays it is used very less, mostly in, I would say, anti-venom therapy. In case of, you make snake bites and then, rabies, that's what I was saying in this. And in case of immunodeficient, there are certain people where the immune system is very deficient, either from birth or usually it's because of some problems during birth. So like they will not have these lymphocytes. In those cases, you give people this intravenous immunoglobulins or antibody, a mixture of different antibodies, because now you have a mixture of antibodies which are against many different infections. So and it will last for, so they are kind of protected for 4 weeks, 3 weeks is what I would say, because then the antibodies are removed from our system. So it's a constant therapy which has to be given to them. But it is one way of where this is used. Okay, so we were talking about T lymphocytes, and I mentioned that there are 2 kinds of T cells, T helper cells and T cytotoxic, and the molecule which distinguishes them is CD4 and CD8. So all these are basically components of the receptor which are there on this TCR or T cell receptor. On the B cell, you have the BCR, which is the B cell receptor. In fact, it is really an antibody molecule which is there on the surface of these cells. So I'll cover more in detail in the adaptive immune lecture, because this is just to give you an introduction that these are the major lymphocytes which are there. And the B cell has different stages. So when it starts to secrete antibody, it no longer has the BCR on the surface. At that stage, it is known as a B cell or a plasma cell. So it looks a little different. It becomes a little bigger, and it has a lot of endoplasmic this reticulum, simply because more and more of this antibody is being made, and protein synthesis occurs in the ER or endoplasmic reticulum. So this kind of expands. So basically, you will see that whatever changes happen in our cells are usually in response to something, and the changes are also based on what that function of that particular cell is and what it needs to do. In this case, it needs to make more proteins or basically more antibodies, so simply the ER machinery becomes bigger. So if you will see in phagocytosis, you will have more phagosomes becoming, developing. Lysosomes are more important in that particular thing. And so T helper cells, we will go this again, as I said, into more details. They are also divided into different subsets, TH1, TH2, TH17, and Tregs. That I will cover in the adaptive immune lecture, because it's an important area, because these different kind of T cells have different functions, and they become, one subset becomes more prominent in one kind of disease, the other becomes more prominent in disease. So there's a lot of interest in compounds and things which can give a particular kind of an immune response. To give you an example, in allergy you have predominantly Th2 kind of an immune response. Somehow, if you can give something which can shift the immune response from Th2 to a different kind, so allergy is taken care of, because Th2 is what causes allergy. So I will go into a little more detail and give you examples of that, and how some of the my lab also does a lot of work on this kind, finding these natural compounds or bacterial components or different things which can modulate this immune response in the way we want. So this is one kind of therapy. It is also used for adjuvants in vaccines, because in TB, you want to make a higher interferon to fight. So what kind of a compound or a thing which you can give, which will amplify your Th1 response, which results in the production of interferon gamma. So that's another, I was talking to one of your faculty, and she's mentioning that you do a lot of natural products, even plant-based or others. So there is anecdotal history that many of these work for this and this. So we kind of have a knowledge, okay, this particular plant or this is effective against this particular disease. But what we don't know is how is it working? What component of it is working? So if you come to know that, it's more easier to patent it or even to sell it, for that matter, to a company, because now it can make into a drug. It cannot take a plant and make it into a drug. Yes, so it's a simple use of the things which are there. I think I have already mentioned that we express these cluster of differentiation molecules. So, the bone marrow is considered the primary lymphoid organ. This is where the immune cells develop, and the stroma in the bone marrow is important for B lymphocytes to develop. Basically, environment of the cell. So fibroblasts and other things which form the architecture of a organ, all play an important role in the function of the cells which are there on top of it. It can be immune cells, it can be other cells, because now people are even realizing that in the cancer, it's not only the cancer but also the microenvironment or the tumor microenvironment, which is the major culprit. And sometimes even the changes are seen much earlier in the microenvironment, the cells in that particular tissue, than in the cancer itself. Because cancer is only visible when the cells have multiplied to a certain mass. Otherwise, how do you know? That's one of the problems in detection of cancer, basically. So this is what I have mentioned, that T cells will develop into the bone marrow, but then they migrate into the thymus, and that's where they achieve full maturity, and that's why they are known as T cells. And this, I think I have a few slides in the adaptive immune lecture, what happens here. This is an important process, because in the thymus is where you get rid of cells which will react to your own self. And secondary lymphoid organs are basically where the cells are supposed to encounter different kind of pathogen, or where the immune response will be initiated. So bone marrow is the organ where cells will be made, and thymus is also one of the organs where the development will take place. But all the other lymph nodes and other organs are where the cell response will be initiated, immune response will be initiated. So these are known as secondary lymphoid organs, and they are connected to each other by the blood and the lymph. So lymph nodes I mentioned, spleen I mentioned, this mucosa associated lymphoid tissue. So these lymph nodes are, distributed all over our body, but in the area of the, like in the nasal canal or alimentary canal, in the lining itself there are patches like this. Especially in the alimentary canal, these are patches which are known as Pear's patches. It's like small lymph nodes in a way, you can say, but it's basically the lining and imaginates like this, then goes, and then here you have pockets of T and B cells just there for, and these are known as mucosa associated lymphoid tissue or MALTs, because you don't want the lymph node to be here if something has to respond here. So cells need to be in the vicinity of that area. Okay, so this finishes just the overview lecture. So now we will start with the innate immunity if you want to start now, or you want a break for 5 minutes, whatever way, it's fine with me. And so the next lecture, how I have organized is, I will tell you how these myeloid cells, basically dendritic cells, macrophages, neutrophils, how they respond to pathogen. What are the different things which will happen? Before that, what are the barrier protections we have? And then I will tell you how we can use it for different things, the knowledge. Okay. Okay, that's fine. That's what I was thinking.
One announcement is there. Lunch is arranged in outside behind beside the department. You can go and lunch.
So now we'll continue with the innate immunity. So go deeper into it basically. And then in this I'll introduce the pathogen recognition receptors. Slowly it will come. And then in the next lecture, which will come after this, is going to be how to use that for making adjuvant and other things basically. So Metchnikoff is the one who's believed to be the father who discovered basically phagocytes. And he realized that there are special cells in the body that can kill microorganisms which are coming in. And this was done with, starfish larvae and other things. But major things is that he realized that there are cells in the body which were capable of taking in foreign substances and destroying it. And he gave I don't know if he gave them the name, but the name phagocytes was coined for these cells. And so this, I think we will, this is just a kind of a recap that there are two kinds of systems. So we will, I think we have not had, I believe, not so much of a gap that we've forgotten what we've learned. So how is the innate immune system organized? So first you have surface barriers, and then you have subsurface protection. So surface barriers include, things like skin, and then also, the cellular organisms which are below the skin form this subcellular protection. Skin is one example of the surface barrier. Your nasal lining, intestinal lining, eye, everything which is exposed or which can be exposed in some way or the other is a barrier, basically, which is preventing something from coming in. And this is, as I mentioned, the vaccination really results in no change in the innate immune response. What it changes is only the adaptive immunity and the memory which comes with it. So when you are given a vaccine, it may initially activate the innate immune system, but whatever remains after that, why our system remembers how to fight something better is because of the adaptive immune system, not because of the innate immune system. Just kind of remember this. So innate or non-specific immunity is considered, it has like two lines of defense. First is the barrier. So in that you have a lot of different components. Intact skin is one. Mucous membranes, because your nasal lining and the intestinal linings all secrete mucus, and which plays a major role in fighting, preventing bacteria and other things from coming in. And the third major component, which has become very important, is the normal microbiota. So all of us know that in our intestine and even in our oral cavity, we harbor a lot of microorganisms, bacteria, which kind of reside within us symbiotically. they also produce a lot of beneficial substances which are good for us, because even the vitamins and others are synthesized by these bacteria. So they also form a major special form of innate immunity, which I will also tell you a little later, because they occupy all these spaces in our body. If these spaces are empty, it will be occupied by all the infectious organisms. It will give place for the infectious organisms to come in. And in the second line of defense is when something breaches these defenses and comes in, then you have cells like natural killer cells, monocytes, macrophages, neutrophils, phagocytic cells, and then there are other responses known as inflammation, fever, and production of antimicrobial substances, which will now fight off whatever has come in. And the third line of defense is the adaptive immunity, which we will talk about a little in another lecture. So this slide kind of summarizes a lot of the barrier functions. So in the skin, you have the epithelial cells, which are joined very tightly. And in the skin, you have a protein known as keratin, which is very, very hard protein. So the layer on your skin is so hard that it is not very easy for something to penetrate it, because the keratin, the cells on the top layer are dead. They have keratin, which is a strong protein which kind of forms a layer on top. So something penetrating that is a little difficult. And apart from that, skin also produces a number of defensins or different kind of peptides, antimicrobial peptides, which can kill things which come on the surface. So that is from the point of view of skin. Then in the airways, you have the airflow in a certain ways. You have cilia and you have mucus. Mucus can trap the organism which is there. And then in the tears, basically in the eyes and nasal cavity, you have the tears and the nasal cilia, which will, and the mucus production, which will help in killing the bacteria. So because in the tears, you have enzymes known as lysozyme, which are present, which is there also in the saliva, which can kill the bacteria. And similarly, in the airways, in the lung, we produce some kind of soap. Surfactant is a mild detergent in a way. So these detergent-like substances dissolve the cell walls of bacteria, and these are produced by our lung cells. And if there's a defect in this, then you get more infections. So it's like different things which are produced by our body, which can, which try to take care of the bacteria even before it is encountered by immune cells. pH is also an important thing. So in the stomach, you have lower pH, very high, like it's highly acidic. So that also kills a number of bacteria, because they cannot survive the low pH. And there are also enzymes known as pepsin, which will digest. And as I said, there are a lot of, in all these places, a lot of antimicrobial peptides are produced. And these are all different kinds of antimicrobial peptides which are produced and which can fight infections. And especially in the airways and alimentary canal, these are all, in fact, more and more numbers are coming up, but these are all produced by these epithelial cells and even neutrophils and other things. And the last is, as I said, histamines are also, histamines and beta defensins are also kind of antimicrobial peptides. See, all defense is good, but in all cases, what you want is the immune response comes up and then it should go down. So in all these cases, like if you take histamines or beta defensins, if they are produced when required and then they come down, it's fine. When they remain there for a long time, or if there's they keep on being produced, then they start causing damage and cause problems. And the last is the microbiological innate system, which is normal microbiota. In fact, now with the new research, it looks as if most of the diseases are controlled by the change in our microbiota. It seems to be linked to our diet, basically changes the microbiota which is there in our thing. And it seems now to be linked even to Alzheimer's and other diseases. In fact, there's a whole initiative where people are trying to map the microbiome to see which classes of bacteria are useful for us, how we can increase those bacteria and decrease the ones which are harmful for us. See, one of the problems with this kind of an approach is that this microbiome is inherited from our mother's side. Okay, whatever your maternal microbiome is there, that is what you inherit. There are only two things which come from your mother's side, definitely. One is the mitochondria and the other is this. So even if you change the microbiome by, say, you have a lot of therapies people talk about where I think you take antibiotics or other things which slowly get rid of all the bacteria you have, and then you repopulate them with the bacteria of your choice, which are beneficial. But within 6 months or so, it has been shown that it goes back to what it was. So it's not a permanent kind of a solution. But it is believed that if you do it many times, then slowly the number of beneficial bacteria will maybe increase from whatever numbers it was there. So that is one thing which is right now highly, at least popular in the US. You will hear about these probiotic foods, even yogurts and other things, which is everywhere nowadays. That basically food seems to control most of the things. So, so we are going away from track, so we'll go back to it. So this is just to show that the epithelium forms so many layers on the top, and it is so tightly connected that it is not easy for something to penetrate. And this is the cells in the respiratory tract and even their reproductive tract. They have these cilia, and then they have these mucus producing cells in between. So mucus and cilia kind of throws the things out, and mucus traps them in many ways. So that's how these things prevent things, bacteria from coming in. So this is a very simple, textbook example that this is your epithelium, and these are your macrophages and dendritic cells. I told you that they are just below the epithelial layer. So if there is an infection, now for whatever reason something is adhering to the epithelium, and then many of the virus bacteria secrete proteins which will dissolve the junction proteins between the epithelium. So now the epithelium becomes open, and now they can come in. So this is what is happening here, and these cells are trying to fight it. And as I said, that they will eat it also, but they will also take part of it and go to the lymph nodes. And here now adaptive immunity is coming into play. So this is a very, how do I say, very simplified version of what is happening really. So what happens when there's defect in this epithelial system? It leads to different diseases, and these are the few I have mentioned here. So in cystic fibrosis, what happens is the mucus which is produced is too thick. So if it is too thick, it is going to form a very thick layer. So people have trouble breathing. It will also congest your airways. That's what happens when you have any kind of influenza or bacterial infection, that the mucus is too much. So you have trouble breathing. So in cystic fibrosis, it is a variation of that. And also the cilia are defective. They don't beat properly. So there is also a separate syndrome where the cilia don't move at all. They are there, just stay there. They don't do their function. So in case of burns, this is, you are more prone to infections simply because your epithelial cells have been burnt away. So that's why they are kept in isolated wards. And so similar to burns, there's a disease known as pemphigus, and in this also the epithelial cells lose their integrity. Basically, the epithelial cell, if it is not intact, then things can come in. So these are some of the defects. So the chemical barriers which I had mentioned. So low pH is also there in the vaginal and the urinary tracts, and definitely in the stomach. And defensins, I have mentioned that these are antimicrobial peptides, and what they do is they insert into the bacterial membrane and form pores. So most of the time, all the other substances, the way they try to kill bacteria is either basically harm their cell membrane or cell wall. That way, whatever contents are there inside will leak out. So the cell will no longer survive, right? That's a very simple way of trying to kill them. And these enzymes, they also degrade. So peptidoglycan is nothing but the cell wall of, it's a component of the cell wall of the bacteria. So most of the enzymes which I mentioned, like pepsin or lysozyme or whatever, there's papain. Papain is known more for antibody, but all these things, they will act on all these different components of the cell wall and dissolve them. So that's when the cell, it becomes vulnerable. It's going to not be able to survive that long. And then interferons are basically cytokines, but they're this chemical barriers because they were discovered very early, and the name interferon came because what they found is that this particular protein was able to interfere with viral replication. So the name interferon came, and as you can see, it's written here that it is produces substance which interfere with viral reproduction. So it's like one of the functions of the interferon, but it does a lot more things. It can act on macrophage activation, and we will go later also into it. T cells produce a lot of this interferon, and these interferons come in which have like different functions. So interferons are important cytokines which especially for viral, and now people think it is playing a major role in cancer also. So this is just to show that in all areas, it's kind of summing up what I have been saying, that there are antimicrobial factors in the saliva. So these are different kind of enzymes which are present, lysozyme, peroxidase, lactoferrin, and in the tears, and so cilia and other things will remove the inhaled particles. And this is even the fatty acids, which are fat, which is their lipids on the skin or sebation, and the sweat which is produced have an anti-defensive action. So all things are important, is what I'm trying to say, whatever our body produces. So it's not really considered a part of the immune system, but it is a part of the first line of defense. So as I said, that they take up spaces so the invading microbes cannot come in. So I'll give you an example. In the laboratory, if you're trying to establish a salmonella model, and if you use mice do not take, if you infect the mice with salmonella, they will not take it. They'll remove it. Like you don't have a model. They don't get infected. So you have to treat the mice first with 5 to 7 days with an antibiotic, streptomycin or whatever it is, to get rid of the normal microflora. So now there's space. Now if you infect, the whole of your gut is now populated with the salmonella, and that's how you get this infection. Similarly, I think even for E. coli, people try to do this, but salmonella, I know for sure that it is done. So it's basically what you're doing with this antibiotics is you're getting rid of the normal flora. So it's creating a space, and now that space can be occupied by whatever organism that comes in. So in this case, salmonella. So that's why if you see when you're during many a times when you take antibiotics, you suffer from intestinal problems. It's because of this, because your normal flora gets disturbed, and so this just tells you that how important this is. And then they also produce a number of compounds that are toxic to other bacteria. And the other thing is that they provide stimulation to the immune system, because we have an immune system in the alimentary canal and in the lung and oral cavity also. They are seeing these bacteria. Even though they are commensals, they activate the immune system in a very low level. So in fact, some people think it is an active thing where the immune system is activated and then gets suppressed, so that it is kind of in the cycle goes on. So the immune system is constantly being stimulated by these cells. And now people have realized that these microbes produce, all these vitamins are produced by whatever we don't synthesize, like the enzyme, biotin, vitamin C. All these are produced by these bacteria, apart from the food which we can take from that, but bacteria also synthesize these compounds. An example is that even the bacteria in our stomach, in our intestine, produce these free fatty acids, short chain fatty acids known as butyric acid and other things. They kind of break down the fat into that, and those are very important for our. So it has been shown that in the intestine, if you want to keep the immune system suppressed, or you don't want to have this Crohn's disease or inflammatory bowel disease where you have a lot of inflammation, these small short chain fatty acids act on the immune system and suppress it. So if you remove the bacteria, then you start getting these diseases also. So there's always an imbalance of this kind of microbiota, which are not producing these free fatty, short chain fatty acids. So they do a lot of different functions, is what I'm trying to say, that break down the fiber into small things which are useful for us. And maybe later we can show you this video, because it kind of summarizes whatever I have shown in a different way. It's a 2-3 minute video. We can see it some other time. So once something has breached and come in to our body, then the first cells which will come in contact, apart from dendritic cells, are the neutrophils and the macrophages. And neutrophils first, because the number of them is so high in the body that the chances are that they are going to be the first cells to come in that area. And what they do is, their major function is to eat, basically, which is phagocytosis. So phagocytosis is the capture and digestion of foreign particles. And then they also produce these chemokines and cytokines, which I mentioned, which will attract other cells to the area where they're required. And side by side, phagocytosis is also activated by opsonins, which I'll mention. These are proteins which are produced by a different system, complement system in our body, and they can attach to microbes. I'll mention it a little later. So this and next 2-3 slides just show you more or less the same thing, that you have a microbe, the cell membrane invaginates, it forms a phagosome. Here is the phagocytic vesicle. And then you have a lysosome in the body which goes and fuses with it. And this you have a number of enzymes which will cause the breakdown of the microorganism which is there in the phagosome. Here you can see, and then it is expelled out. Okay, this is what happens mostly in the neutrophils. It's a very common thing. In macrophages and dendritic cells, there's a little bit of a diversion, which I'll show you a little later when we come to it. So here also it's different ways of depicting the same thing, that how the pH is changing. In the phagosome, the pH is still like 6.2 or more near 7. And in the lysosome, because it's highly acidic and the enzymes are there, that you have this breakdown and the digestion. So how does our body recognize the microbes? That's where they are recognized by special receptors on the phagocytes, and they can recognize the microbes because they have a cell wall, and we don't have that cell wall. Our cells in the body are all with cell membrane. We don't have a cell wall on top of it. Most of the bacteria and viruses or whatever, they always have a cell wall, and this cell wall is made up of usually lipid, protein, and carbohydrate polymers in different combinations. The structure can be different, and that is what gives them, that is what makes them unique. And for that, I have a whole part of it devoted to it. So that's why I'll come to it later. And the phagocytes, they are digesting, but they're also producing a number of cytokines, chemokines, which leads to inflammatory response, production of antimicrobial peptides, etc. So, and this, to a certain extent, I had already mentioned that if you have a chemokine concentration, this is very high, it kind of allows the cells to migrate to the area where the chemokine concentration is high, attracting of, basically that's why it's known as chemo attraction. attracts the cells to the site of injury and affects nerve cells. This is basically inflammation which is happening, which is, inflammation is a coordinated response to tissue damage. So there are 5 major signs of inflammation, which you have seen. If even a mosquito bites, you will see that the area becomes red, a little bit, this is all inflammation because something foreign has gone in and your cells have come there to react. So if the cells come there, because the area is small, it will become a little swollen, and cells may produce heat because the cells are accumulating, they are dying, their pain is also there. Nerve endings have become more, because of the chemokines, cytokines secreted, they have become more sensitive. And so basically the inflammation plays a major role in containing sites of damage and restore tissue function. It's only when inflammation, we hear a lot about how inflammation is the cause of most diseases which are there. Inflammation per se is really required. It's only when it is uncontrolled that it leads to problems. In all cases, whatever functions you have, if they are not controlled, they lead to problems. So this is just to show what happens in an inflammatory response. Basically, capillary, because cells need to reach there. So capillaries have to become more permeable to allow the cells to reach there. So that's what happens. And this is what I had mentioned, that the area becomes swollen red. Temperature increases from increased blood flow because cells are moving fast. But there is also another reason. The cytokines, some of the cytokines which are secreted are pyrogens. They will act on the hypothalamus to increase the temperature. So it's not only because of blood flow. And so basically there is ultimately cleanup of pathogens. This is just depiction of the same thing, that you have, cut in your knife. You can have bacteria which can come in via the cut. So you have tissue damage. All these things which are there in the vicinity of this will become activated to try to. So see, so now all these chemicals are being produced, histamine, prostaglandins, and by the cells which are damaged. But side by side, you also want to make sure that the barrier is closed as fast as possible. That's why you have blood clot being formed. And the capillaries are becoming dilated for other cells to come in to fight whatever has come in, the blue bacteria, you can say. So both the processes go on simultaneously. So I think it's more or less showing you that from the blood vessels, how cells are coming in. So they can kind of squeeze through. This process is known as diapdesis. So that there's a whole way how cells can squeeze through the capillary and go into the tissues. It's a whole area where people are studying how it can really do that. But in short, cells squeeze through and go through the tissues. So ultimately, the final thing is that the scab is formed, and then the bacteria has been removed. So resolution of inflammation has taken place. Tissue, epidermis, which was destroyed, is regenerated. So if you have a problem in any of these processes which is happening, it's going to lead to some kind of a disease or the other. So it's more important to understand the process itself and realize that where you can intervene. That's how you can think of therapies. So this is, as I said, more or less the same thing as what I'm saying, that tissue repair is promoted once everything is taken care of. So we usually consider fever as one of the problems, but fever is one way of your body is telling you that there's something wrong with your something is wrong and you should now take care of it. So that's why inflammatory response is often accompanied by fever. One of the other, because of certain cytokines which I told you are known as pyrogens. And one of the advantages of fever is that many of the pathogens can multiply or function well at 37, which is your normal temperature. But when the temperature rises, many of them cannot multiply, or they will die. So that's also one way of, and also the higher temperature activates, everything in your body now functions at a faster level, because more energy is in a way being provided, more is generated. So fever is also important in that sense. So apart from all cytokines, chemokines, there's also a system known as complement system, and these are proteins which are circulating in our blood and the fluids which which are basically just circulating. And what happens is when they detect the presence of a foreign material, it's known as complement cascade. Immune cells by themselves don't increase. What they do is they secrete certain. So one is things start moving. They're moving faster because of that. Body temperature is raised. But there are certain cytokines. You take things like IL-1 beta or IL-1 alpha. These are known as pyrogenic. TNF alpha. These are known as pyrogenic. They will go and act in the hypothalamus and tell the hypothalamus to raise the temperature. So when the innate immune cells get activated, they will secrete these cytokines. And these cytokines will then go to the hypothalamus and tell it to activate. That I don't know. It has a thermostat. Hmm. Very thick. No, mucus is produced maybe in the same amount, but there is a certain consistency of the mucus, right? It should flow. If it is not able to flow, then it is going to form a thick blob there, right? So it is not going to allow air to also pass through. So that is what the problem. So that's why these people have trouble breathing.
Because right, it is directly acid is attacking the.
So there is not a single mechanism all the time, no? Otherwise, it would have been very easy to develop a therapy. In cystic fibrosis, what they realize is this mucus which is produced, why is it so thick? Because there's a change in a gene mutation. One of the reasons is. So now they're trying to do a therapy, making a gene therapy for that gene, so that maybe you can introduce into the epithelium and now the mucus becomes normal. That's one approach which is being tried. So first you have to identify why is it happening. In the ulcer, okay, stomach lining is done, but that is a much later reaction. What is the initial part of it? Why is it? Why is the ulcer even forming? Is it because of food you eat, or is it because there was an injury there?
H. pylori.
Yeah. So H. pylori is one of the reasons, right? Stress is also another reason which leads to ulcer. So in all cases, then you have to think about how you can. If you can find one mechanism, it will be much easier. So complement proteins there are approximately 30 proteins basically, and they work by many different methods. And this you will find in any textbook, so you can go and read. But the major thing is that they create. See, all of these I told you that ultimately they work in different, but they try to create pores in the membrane of the foreign cell, so it dies. That's how most things can kill, because once you have destroyed the integrity of this membrane, cell will die. There is no way. But the other major thing which the complement also does is, if it cannot kill the bacteria, it also attaches to the bacteria. This is like activated complement proteins. And these activated complement proteins which are on the surface of the bacteria are kind of like flags. It's like, okay, there is a fluorescent flag on these. It's not fluorescent, but I'm giving you that those cells, those bacteria now have something which will tell the immune system that please remove these. It's like a flag that, okay, something is there, you have to remove it. Signal is there. So there are two ways. One is they will directly attack the bacteria. The other is that they will also mark the bacteria for killing. Complements also activate, the other cells of the innate immune system. There's a whole, as I said, the whole textbook can be dedicated to that. So we will leave it that. This is just a cartoon which depicts that how the bacteria says that I'm hit, now I'll be removed. And the You know, I told you that the complement proteins bind the bacteria. These are C3b component of the complement. And this process is known as opsonization, because this kind of, these marks which are there tell the phagocytes that you have to remove them, because they have a receptor for this, the CR1. It recognizes this component. So if this is there, it will be taken up by these phagocytes. It can be neutrophils, monocytes, any other cells which express this CR1, and it will be killed. So basically, it's, our immune system is like any other army. We even if to do the same function, you have different mechanisms how to try, because you cannot depend on one thing. If that fails, then what happens? So that's why you have multiple things in play which will all act together. And also there's a lot of redundancy, that if this doesn't work, then this will take care of it. You know, it's not like, okay, one thing is not working, now we are very susceptible. You have layers and layers of it. The other things will become more prominent in those. So to give you an example, I was telling you that dendritic cells are very important, that they initiate the immune response. But if you make mice lacking in dendritic cells, it's not that they are highly susceptible to infections. Maybe a little bit more, but then monocytes and other cells also take over the function. So very few cells are such that if you remove them, then it is going to always result in a disease. First of all, it has to be, the deficiency has to be such an extent that it is like total. Then only it leads to some kind of a problem. More is the problem in the adaptive immune system than in the innate immune system. Innate immune system, because all these cells can phagocytose, you know, like neutrophils, monocytes. Other cells of the body also can phagocytose, maybe not as efficiently as these. But if there's a missing component, other cells will try to take over. So maybe not, as I said, it's not as efficient, but it does work. So now we move to the next portion, is how we recognize that something is foreign. Okay, because innate immune system sees something round. It doesn't mean that it is a bacteria, it can be our own cell. So as I mentioned, one of the things which is different between the microbes and us is that the cell wall which is there on there. And this cell wall are mostly made up of lipid proteins and lipid carbohydrates, which form repeating. These are usually polymers. So it's the chemical structure of these polymers which is recognized by our cells. It's not the microbe itself which is recognized by our cells. And these patterns are recognized by different receptors on our cells. And these now as known as pattern recognition receptors, because they recognize these patterns of the chemical, you know, the structure basically. They don't care if you give the structure separately. Without the bacteria, it will still recognize it. But that's why these whole array of these receptors is now known as pattern recognition receptors. And why is it important is it tells you that you don't need a whole organism to generate an immune response. You can take a part of the bacteria or a fungi or for anything, for that matter, anything, and you can generate an immune response. So this is what led to the thinking of that it can be used for therapy. But that, as I said, will come later. First, we understand what happens. So there are 4 major classes of these PRRs, or pattern recognition receptors. First is toll-like receptors. Then you have C-type lectin receptors. And then this is RIG1-like receptor and NOD-like receptors. And if you see TLRs, CLRs, RLRs, and NLRs. So in all these cases, when they are engaged by the microbe, you will have a signaling response, signaling pathway which is activated, and then innate inflammatory response. So now we will go one by one into each one of those. So this is it's a more of a TLR receptor. So this, don't worry about the structure of it, but basically it has a signaling domain which is known as a TIR domain in all cases. Okay, these are different TLRs, TLR2, TLR1. Here what we are showing is that sometimes the TLRs come together for functioning. Like TLR2, it usually has to work with another TLR to function, and it recognizes lipopeptide. So I'll show you in the next slide simply. I think it will be easier. So TLRs recognize many types of pathogen molecules, and the way they were discovered was in the fruit fly, there's a receptor known as toll, which does the same function. And when some scientists deleted this toll from these fruit fly, these fruit flies became became highly susceptible to fungal infections. In fact, you can see them covered with fungus the moment this was deleted. So that's how and when they were discovered in human, they were found to be analogous to this protein. So that's why the name toll-like receptors came. It's not like so these toll-like receptors not only recognize, PAMS is pathogen associated molecular patterns, the patterns which I was talking about. So pathogen associated molecular patterns. And DAMs, so now what people have realized is they not only recognize foreign substances, they also recognize many of our own substances which are produced. And these are only produced in our body when there is a damage. So they are known as damage associated, or of the damage associated, some people say danger associated molecular patterns. So these molecules or these proteins which are produced in our body are dangerous to us. So they are also recognized by toll-like receptors, but the name is DAMs in this case. So this is, okay, I think the slide is not big, but I will explain. So you have about 10 toll-like receptors in humans. So this is just to show that, okay, maybe I'll show you here. So toll-like receptor 4 is what you know very well. So I'll go with that. This is 4 basically. So toll-like receptor 4, you know, lipopolysaccharide, all of us know. We use a lot in any kind of experiments we do. This is a component of E. coli bacteria or gram-negative or gram-positive bacteria. I don't remember which one. But it is recognized by this TLR4. And the peptidoglycan, which we showed you, is recognized by TLR2 and 6, which is shown here. In fact, 2 and 6 also recognize a number of fungi. And there's TLR5, which will recognize the flagella on the bacteria. So, and that's why these toll-like receptors are also expressed in different areas of the body. Now, TLR5, because it recognizes bacterial flagella, it is expressed more in the cells of the mucosal system, like in the alimentary canal and in the nasal, because most of the bacteria which come in via these routes have flagella. So you need to have something which recognizes the flagella. And these TLRs are not only present on immune cells. Now people have found that epithelial cells also express a number of these toll-like receptors, because those are the first cells, if you see, which are going to come in contact with these microbes. So the bottom line is that all these, we have different TLRs, and all of them will recognize different kind of molecular patterns, because all bacteria do not have a same kind of cell wall. Some will have lipid, even LPS for that matter, comes in many different flavors. Like if you take E. coli LPS, it has a different kind of lipid tail. I don't think I have that slide here, but if you take LPS from gingivalis, P. gingivalis, it has a different kind of lipid tail. And because of that, both of them bind different receptors. E. coli LPS will bind TLR4, while P. gingivalis will bind more TLR2. And the response is also very different. The TLR4 E. coli will give you very high immune inflammatory cytokine secretion, while P. gingivalis will give you very low. So it's how which receptor it is being engaged, the response also changes, the immune response. Like if there's a fungi, if it will bind, it results in a different kind of immune cytokine secretion. And this knowledge that how a bacteria will bind and what kind of a response it will give, because this response which is generated now will decide what kind of an adaptive immune response you get. So in a way, how you are activating the immune system is telling you the end product, what will happen. Will you get more memory, less memory? What kind of a memory you will get? What kind of an immune response you will get? So if you can manipulate this first part, you can basically manipulate the immune response, right? How you are activating the immune system. If you can understand, if I activate it this way, I can get this. If I activate it this way, I can get this. So in this disease, if this is predominant, can I change it now to the opposite, which is maybe beneficial for me? So that's why this knowledge of toll-like receptors and everything, how the pattern recognition receptors are activated is very, very important. Even for vaccines, many times when you take, if you take the whole organism vaccines, then they are very successful. Like if you take, this, for, like I'll give you an example. In the hepatitis, you give only one protein. It's a subunit vaccine. And that immune response against that protein is sufficient for you to provide protection. But in case of MMR or influenza and all, you have to give the whole organism in a way, right? So the only thing is this organism is not infective. So, but it also leads to a number of side effects and problems which are there, because there's a chance, now if you take polio virus, there's a chance that 1 in 10,000 people it reverts back and it causes paralysis, right? So that's why people want to move away from all these whole organism vaccines, and they want to find components of the bacteria which can elicit an immune response, which can provide protection without causing infection. So, but major problems which come with it is when you reduce it into a small protein, it is no longer highly immunogenic. It does not induce a very good immune response. So then you want to combine it with something which will induce a good immune response and which may help in providing better protection. In that, people are using a lot of these toll-like receptor ligands in a way. Like you cannot use LPS, but they have found out that in the LPS there is a lipid A moiety, which is what is really activating it. So now that also causes a lot of fever, inflammation. So they've modified it in such a way to identify the active component and trying to use that as an adjuvant or vaccine to activate the immune system. Example is cancer. The immune system is very suppressed in that. So you're trying to find compounds which will break the tolerance. So that's how you can use these toll-like, these pattern recognition receptor ligands. But first you have to know that which is the ligand which will activate, which receptor, what kind of immune response it gives. Is it going to be beneficial? So that whole study needs to be done first before you wish to use it, right? So that's what, and these toll-like receptors and all are all kind of newer discoveries in the year. They are from 2000 onwards, you can say. So they're still new in the sense we are still in the learning phase, even though many of them have moved to the clinical trials also. But so this is just to show you that how TLR1 can recognize triacyl lipopeptides, which includes mycobacteria and gram-negative bacteria. And TLR2 recognizes peptidoglycans, which are major component of gram-positive bacteria. They also recognize trypanosomes, and lipomannan is also a component of mycobacteria. Zymosan is part of yeast cell wall. And then you come to another TLR, which is TLR3. It recognizes RNA or double-stranded RNA. So viruses usually do not have the machinery to multiply on their own. They utilize our machinery. So they have to infect us. So then what is foreign in them? Their capsid, but their nucleic acid is what, because they are going to release that to multiply, right? So that's why we have these TLRs which can recognize these different kind of nucleic acids which are present in viruses and bacteria for that matter, but mostly viruses. So the TLR5, as I said, flagellin earlier, it's bacteria. And then you have TLR6, So TLR6 usually functions with TLR2. That's why you will see that zymosan and some of the things are common between the two. That mycobacteria, gram-positive, and yeast and other fungi. 7 and 8 are also recognize different kind of nucleic acids from viruses. And there's also TLR9. Yes, that's what I. So basically these toll-like receptors, the nucleic acids which they recognize from virus and bacteria, they recognize what is different from our nucleic acids. So usually we don't have single-stranded RNA in our things, or we won't have, which is what is recognized here. Single-stranded RNA viruses are like influenza viruses. So these kind of nucleic acids are not present in our own body. So that's why they are foreign to us. So double-stranded RNA is also not present. The genome of these viruses is unique in that sense. So that's why it is recognized as foreign in our body. And there are other reasons why we are usually not, these TLRs do not have access to our own nucleic acids also. And that is also another reason they don't recognize. TLR9 recognizes these CpG unmethylated sequences. So these sequences are numerous in bacterial DNA and much less in our DNA. So that's another thing which is foreign. So that's why it is able to recognize those. And these I will not worry, because some are, like this TLR11 I think is more in mice than in human. So I will go till TLR9. 10 nobody knows what it recognizes, so we will not worry about it. So what happens when toll-like receptor bind to PAMPs? So as with any receptor, you will have activation of signaling molecules. So this is just an example of signaling, that if the TLR1 and 2 are coming together, you have an adapter molecule known as MyD88, myeloid differentiation factor at 88. This is important because this is common downstream of all toll-like receptors. So people have made mice which are deficient in this, and then they realized that this whole signaling molecule cascade does not proceed. So that's why this molecule is kind of very important. And these are IRACs, which are all, these are, basically this is like any other signaling cascade. Anytime a signaling occurs in a receptor, there's going to be something binding to a receptor, then you have adapter molecules, and then the signal proceeds with different proteins becoming phosphorylated, dephosphorylated, phosphorylated, dephosphorylated, and that's how in any thing, these components will be different with different receptors, okay? The adapter molecule will be different, the molecules which are associated will be different, but the major mechanism is more or less the same. Phosphorylation will occur because you require energy, which will come from ATP, and that's how basically the signal moves. This becomes phosphorylated, then this phosphorylation will be transferred to this, and ultimately you have an activation of a whole major transcription factor, which can be, this is MAP kinase pathway, so you have AP1, otherwise this pathway leads to NF-kappa B. So there are other transcription factors also, but as I said, in most cases, you will ultimately, this phosphorylation leads to activation of a transcription factor, which will then go and bind to the DNA and initiate the transcription of various genes, which will secrete whatever is, whatever signal has been given by the receptor. In this case, it is mostly inflammatory cytokines, chemokines, etc. So NF-kappa B is a major transcription factor which is involved in inflammation. In fact, a number of drugs and things have been tried to be developed against this. But one of the problems is that NF-kappa B is a very ubiquitous kind of transcription factor, which is not only downstream of toll-like receptor pathway, but downstream of many, many signaling pathways. Like the pathways will get activated in different ways, but ultimately it is going to be NF-kappa B. So that's why drugs against this have not been very, very successful, because you have to choose your target also in such a way that it has to be unique. It should not affect other functions of the body. Because, okay, it's killing the bacteria, but it's also killing you, then it's no use, right? You have to. So, toll-like receptors are not on the surface, they are intercellular. So all the ones which recognizes nucleic acid are not on the surface, because how will they encounter nucleic acid on the surface? It's not the bacteria, breaking apart, and so that's why in all these cases, these are inside the endosome. That's where the bacteria will be phagocytosed or the virus will go in. So that's where the most chances of the nucleic acid coming in contact with the toll-like receptor is going to be there. So these are all endogenous toll-like receptors. TLR3, TLR7, and TLR8. TLR7 and 8 are more or less similar. It's that they are found in different kind of cells. Otherwise, more or less they function in the same way. And so in these cases, when the other TLRs are engaged, NF-kappa B is activated and it leads to production of inflammatory cytokines. Here also the same thing happens. You see, in this case, also MyD88 is there. Only this particular toll-like receptor has different molecules, the TRIF and TRAF3. But ultimately, it also leads to activation of these interferon response factor 3, 7. But what I wanted to say is, when you have these nucleic acid activating the toll-like receptor, the major proteins which are produced are other cytokines, chemokines, but mostly interferons. And these because you see, one of the things which I told you about interferons is that interfere with viral replication. And here, what are they fighting? They are fighting virus infection, because all these are nucleic acids which are from viruses or bacteria. So that's why they produce very high levels of interferon, because the purpose is to block the replication of viruses. So that's why the signal which is given tells them what to produce. So in this case, interferons are going to be more effective, more interferons are produced. In this case, inflammatory cytokines are more required, so more inflammatory cytokines are produced. So now in this way, suppose you need to produce interferon in your body for maybe fighting viral infections, then you know that you need to engage these toll-like receptors. And how do you engage these toll-like receptors? You need to have these kind of nucleic acids. You can even synthesize single-stranded RNA, and it should be able to produce the same effect. And that's what people do in that. So it's kind of one way of using it for therapy. So that was toll-like receptor. So on our surface of our cell, so it's not only that viruses, fungi will come and that's all we need to recognize, because the microbes are equally important. They also modify their cell surface in such a way that now they should not be able to be recognized by toll-like receptors. Some have only carbohydrates on their surface, or the kind of carbohydrates on their surface. So you have another class of pattern recognition receptors known as C-type lectin receptors. So they also function in a similar way. As I said, in each cases, the downstream molecules will be different, but ultimately, what gets activated is the NF-kappa B pathway and inflammatory cytokines, because that's what is required to fight the infections, right? But the C-type lectin receptors will recognize mostly carbohydrate components. That is the difference. Those will recognize more peptidoglycan, polymers, different kind. These will not recognize proteins. They will recognize mostly carbohydrates. And many fungi have more carbohydrate-like structures. Even if you take, so if you know Streptococcus pneumonia, it has more carbohydrates on their surface. Even your vaccine, which is given for elderly, is a mixture of 23 carbohydrate molecules. Yeah, which is, and carbohydrates are very less immunogenic also. But these receptors recognize carbohydrate molecules on the surface. So, and the third class which we had talked about is the NLR receptors, or NOD-like receptors, nucleotide oligomerization domain receptors. So these are very important. NOD1 and NOD2 have become now very, very important. In fact, I would say that last 5 to 10 years, so much of work has been done on all these nod-like receptors. And what people have realized is that apart from recognizing all these foreign substances like muramyl dipeptides, bacterial cell walls, it seems to be more important in recognizing a number of danger associated molecular patterns in our body. So anytime you have a crystal, like overproduction of something in our body, like cholesterol or uric acid, it precipitates and forms crystals, right? And that crystals cause various diseases, like uric acid accumulation will cause gout. That's what it does. It accumulates on the joint, and the inflammation which you see is because of this uric acid crystals. So these uric acid crystals, basically what they do is they go and activate these nodes, or nod-like receptors, or NLRs. They're also known as by another name nowadays, which is more popular, inflammasomes. And it causes a major, very high-level inflammatory reaction. It leads to production of IL-1 beta, and side by side, the other thing it does is it kills the cell. So there's a lot of cell death and a lot of production of this IL-1 beta. IL-1 beta is a highly pyrogenic cytokine, so it will create fever, but not always fever, but it leads to a number of highly inflammatory reactions in the body. In fact, even for metabolic syndromes, type 2 diabetes, on all these cases now people are thinking that inflammasome is the major pathway which gets activated. See, the reason, so I told you that the discovery started in 2000. So initially, toll-like receptors were the ones which were discovered. So it took like 5 to 10 years to understand how they function, because each time, this was mostly by a Japanese group, Akira. He made a lot of these knockout mice. He made knockout mice of nearly all the components in the signaling pathway. And that's how we came to know, okay, this comes after this comes after this, and this is important for cytokine production, and this is not. So, but making of a knockout mice takes 2 years or so for it to be successful, right? So that's, I would say, the early thousands went into learning about toll-like receptors. Nobody really cared about other receptors. Then they came to know that there is also receptors known as C-type lectin receptors. But mostly it was more from a point of view of fungal infections, so less, they were not highly activatory, these C-type lectin receptors. So I would say that still a lot is supposed to be known for this C-type lectin receptors. And now, for the last 5 to 7 years, a lot of research has focused on these node-like receptors, especially the inflammasome. And it is becoming more and more important because they're realizing that it is associated with a number of diseases. So this, every day they are finding new signaling pathways, new molecules which they think they can target to maybe suppress this pathway. So this is just to tell you that how many different kind of receptors have been discovered. So one of the other things which these nod-like receptors do is they also bind a number of bacterial and viral double-stranded DNA. So this is AIM2 receptors, and in all these cases, the inflammasome makes IL-1 beta. This is not very, it's too down, but it results in the production of IL-1 beta and sometimes interferons also. Now we come to the fourth class, which is RIG-like receptors. So these are also somewhat like TLR3/7, that they will also recognize viral double-stranded RNA. It's simply that they recognize, so all these are there. Some are redundant also, that, recognizing similar kind of structures, having a similar kind of production of interferons. But they, in addition, they recognize maybe some variation of that particular nucleic acid molecule. And also, I think they are there to function together to amplify the immune response also. So RIG-like receptors basically function in very much a similar way as TLR3 and 7. But they also produce interferons in response to viral double-stranded, molecule. And this is So activation of these PRR signaling molecules leads to a large variety of genes. As I mentioned, all these things will be produced: antimicrobial peptides, type 1 interferons, cytokines, chemokines, and even enzymes like this INOS pathway and COX2. INOS it leads to production of nitrogen oxide and other things. These are also killing of, they can also kill microbes. Even reactive oxygen species, I think I have it maybe here or not, I'll see. That also kills. See, reactive oxygen species lead to production of hypochlorite or bleach-like things and superoxide H2O2 inside these phagocytes in macrophages and in neutrophils. So hydrogen peroxide is highly cytotoxic, highly killing to bacteria. It is one way you disinfect things, right? So that's why, and it's produced in our cells. Similarly, bleach is also produced, not at the level you use, but it also produced in our cells with this reactive oxygen species, and it also, that's why it kills the bacteria. So this, I think some of these slides I have made that if anyone needs, they can use this and you will know what I've talked about for the past, it kind of summarizes a lot of things into small. So here, the only thing I will say is that we have all these different kind of cytokines, IL-1, IL-6, all of them have slightly different functions. Like IL-1 beta, this is it. Here it's written IL-1. It includes IL-1 alpha and beta. Beta is supposed to be more active. So it enhances activity, promotes neutrophil production, vascular permeability, fever, as I said. IL-6 will regulate activity, but it does not promote neutrophil production. It promotes hematopoiesis, activation, like production of other cells, not only neutrophils. So all of these cytokines have slightly different functions. Some of the functions are overlapping, some are different. So like TNF, one of the special things is that it's cytotoxic for many tumor cells. TNF, that's why it's known as tumor necrosis factor. It's basically causing killing of cancer cells. Right? So GM-CSF, granulocyte, macrophage, colony stimulating factor. So it stimulates hematopoiesis, as the name suggests. So like these IL-12 and IL-18, which are produced, these will, when they are produced by antigen presenting cells, they will lead to production of interferon gamma by T cells or a Th1 response, which I will come to later. So what I'm trying to say is the different cytokines which are produced also have different functions. Like all these cytokines I've shown here are all inflammatory, but IL-10 is an anti-inflammatory cytokine. So whenever you have production of pro-inflammatory cytokines, to balance, you will also have production of anti-inflammatory cytokine, because you need to always shut down the immune response when it occurs. So with age, what we find is that this response seems to go down, this IL-10. In fact, we find in our lab, we have seen that the IL-10 production and even the response of cells to IL-10 goes down. That's why there's an ongoing, always not very high level, but low level chronic inflammation in aging people, and which is a major problem. Why? That's what we are trying to find out, because there are many ways why IL-10 can be produced, right? So sometimes it is that how the inflammation is regulated inside that particular cell, that is not, that pathway is not getting activated. So IL-10 is not being produced. So the problem is the IL-10 is also not produced in response. There's not one pathway which results in production of IL-10. So, okay, we find that, okay, this pathway is reduced. But what happens, why is not the other pathway taking care of it? There's other ways of producing the same cytokine. So there is, as I said, there is no one answer. If it was so easy, people would have answered it. So it's simply, with age, I think many of the pathways become dysregulated. It's not that they they don't function in the same way they should function. That either they will not shut off properly or they will not get activated properly. In many cases, immune cells, what we find is that there is a basal level of activation. Like if a young cell is here in activation, the aged cell is here. But when infection is coming in, the young cell is going here, the aged cell is moving here. So it is activated. But when it requires to be activated, it's not getting activated. Maybe it's already so tired by that time because it's constantly activated. It doesn't go beyond that. There is many different reasons for it. So the type 1 interferons. So interferons are three different kinds: type 1, type 2, type 3. Type 1 and 2 are more produced by innate immune cells. What I showed you in response to all the toll-like receptors, they will produce type 1 and type 2, type 1 and type 3 interferons. 2 is the interferon gamma, which is produced mostly by T cells and not by monocytes, macrophages. But you need not worry about it. All of them, the type 1 interferons are more for viral, they are have more potent antiviral effects because they activate all these different genes, and these are known as interferon response genes, and they are very important to fight viral infections. In fact, in some cases, people look at all these genes, and if they are down, then it seems that the person is not going to be able to fight infections that well. So this is just to show what happens. So they can move to they have an autocrine action, they can act on the same cell, but they also go to the other cell and activate these antiviral genes to make them resistant to viruses. So this is, what I showed you earlier, it's more or less the same thing which is happening. So now here you are binding to the PRRs and all this pathway is getting activated. So what we are focused here on the last part of the lecture is how this binding occurs and how. So now the next part we will focus is, now we have learned that there are 4 different classes of receptors, okay, and what they bind. Now the question is how to utilize it for so this knowledge is basic knowledge, right? Okay, this is good that we know, but how to use this for our own, for therapy is what is important. So this is, I think I have mentioned. So there are different feedback mechanisms. If they are not functioning, you will have uncontrolled inflammation. If the negative feedback is too active, you will not have inflammation at all. But this positive and negative feedback mechanisms do vary in different cells. They vary in many different ways also, depends on the kind of response pathway which is activated. The feedback mechanism can be different. So you have to study each one separately. It's not that this feedback is negative, so it will function and it won't work in all cells the same way. Because cells will also not respond in the same way. Some will not have this TLR receptor, some will have other TLR receptors, so they are going to respond differently. So here I'm going to just give you a few examples of how pathogens have also evolved strategies to circumvent all these different mechanisms that we have in place in the innate immune system. See, we are evolving, but the pathogens are also constantly evolving. And if you see, they are evolving faster than us because their reproduction is much faster than us. So that's how they have survived for so long. And so in many cases, you have flagella which is either absent or it is mutated in such a way that now TLR5 cannot recognize it. Examples are Heliobacter, Coxiella, Legionella. All these have altered LPS. So the TLR4 is not being recognized. The TLR4 is unable to recognize this LPS. So the bacteria go undetected. So similarly, HTLV virus has P30 protein, which basically inhibits the expression of TLR4. So the TLR itself is degraded. So again, the it's not going to be, the virus or this particular protein is not recognized by the TLR4. Then several of the Ebola, influenza, vaccinia, they produce proteins that bind the toll-like receptor or viral double-stranded DNA. So now the toll-like receptors do not have the viral double-stranded DNA to recognize because it's already bound by some other protein. So that's one way. So similarly, many of these produce actively proteins which can prevent the activation of pathways. This is like vaccinia virus protein. This A46R will block the TIR domain that blocks MyD88. The one of the major adapter proteins which I told you. So the TLR will no longer function. Similarly, several viruses bind this interferon response factors. So interferon is no longer produced. This example is like West Nile virus NS1 protein, which inhibits. So what it does is all the transcription factors which are activated have to go to the nucleus to bind to the nucleus to activate transcription. So this virus basically is preventing, it binds the transcription factor in the cytosol and prevents them from going to the nucleus. So what this simply is showing is there are so many different mechanisms which are there that this Yersinia bacteria, which is basically the plague bacteria, it inhibits the inflammasome activity. And inflammasome is maybe very important to fight this plague infection. So usually the bacteria always target the pathway which harms it the most. So here, in many of the bacteria are also very smart. So one of the things which is happening is when you have phagocytosis, you have to have the phagosome being formed and then it has to fuse with the lysosome where all the enzymes and low pHs, so that is what is going to kill the bacteria. So in case of Listeria, it ruptures the phagosome membrane. So the phagosome is formed, but the bacteria escapes from the phagosome. So even if the phagosome fuses with the lysosome, there's nothing to kill. The bacteria is already outside in the cytoplasm. So that's how it survives. Then there is the tuberculosis bacteria. It basically, it remains in the phagosome, but it prevents its fusion from the lysosome. It will not allow it to fuse with the lysosome. So the phagosome remains, lysosome remains, but it never fuses. It's one way the mycobacteria survives in fact, mycobacteria survives in the macrophage only, where the phagocytic activity is the most, because it prevents this fusion and it remains there and it's inside the cell, so it's no longer detectable. Also, it doesn't multiply for a long periods of time, so it remains dormant. So there's no activity, no change in the cell that NK cell can come and kill it. That's how it's like one of the mechanisms how it survives for such a long time. So these are just examples. So vaccinia virus, basically it makes a protein. So the cell produces type 1 interferons, but it binds the type 1 interferons. So interferons are produced, but they are no longer able to act or tell other cells. Yeah.
Of latent bacteria.
Yes.
You take a latent MTB bacteria, lyse the MTB bacteria, and give it to dendritic cells and put it back to the patient. Will it help?
So what you're saying is a very simple, seems very simple. First of all, for this is dendritic cell based therapy has been tried for cancer. It is a very expensive therapy because you have to take out cell from that patient. You have to culture it in vitro for 7 to 8 days. You have to load it with the bacteria and you have to put it back. Everything has to be totally sterile, right? Then it has to go back into the patient and it has to work in one time. Otherwise, you have to do this whole process again. So, there was a dendritic cell based cancer vaccine made, given to the patient. It used to cost $90,000 per person. So that's why this approach is not a very viable approach as such. Instead, what people are trying to do is, why can't you do it in situ? Like, why can't you devise ways where you can deliver this lysate or this, what you're talking about, in vivo inside the body using some kind of a nano
But bacteria is not allowing to function properly. No,
But
it is.
you have already killed the bacteria, right? You are saying you are making a latent.
Yeah,
Huh.
yeah. See, the MTB is hiding for decades,
Yes.
and it is coming back and then causing severity.
Yeah.
But you take the make a latent, you do hypoxic culture. You do make a latent TB, lyse the protein, then educate the dendritic cells and put it back.
So, but that's what I'm saying. If you're making a lysate of the protein, why can't you deliver it with a nano particle or targeted delivery to dendritic cells in the body?
But they say that if you give nanoparticle, it cannot be filtered out through.
No, you take degradable bioparticle, right? Nanoparticle. You can make degradable nanoparticle. You can even conjugate it to another adjuvant or something, which will target it to dendritic cells. Or you can take a protein DEC205 or some other molecule which is expressed on dendritic cells. Why do you need to take out the dendritic? Because that adds the whole element of this
more expense.
expense. So that's why more focus is on how you can deliver things in the body with a targeted delivery. And the advantage of monocytes and dendritic cells is these are phagocytic cells. So if you are giving any kind of nanoparticle, it is going to be first cleared up by neutrophils, monocytes, and dendritic cells. So sometimes you don't even need to have a protein on the cell. It will be taken up by this system first. Reticular endothelial system is the first which will clear most of the anything particulate or anything which you are giving as a foreign nanoparticle. In fact, a lot of research was done long time back, even with liposomes and things like that, right? Those are biodegradable. In this case, you are talking about one thing. Liposomes are lipid vesicles, right? Made of. So you can, people have even made mycobacterial lipid vesicles, lipid from so because then it will initiate some kind of immune response which is against mycobacteria. So I would say that it is better to think of ways where you can target in vivo. Then most of the cellular therapies which have failed have failed because of this
Expense.
Yes, because you need CGMP facilities. It has to be, those facilities itself are so expensive. All this process is very expensive. So it is, ultimately, the benefits do not outweigh the cost. Like this cancer vaccine I'm talking about, it resulted in an extension of 3 to 4 months survival. So how many people can afford to give $90,000 to survive for 3 to 4 months extra? It's not. So that's you have to see, for a therapy to move forward, it has to be both
viable.
Viable. That's why. So these are all different examples of how different viruses and bacteria have evolved different mechanisms to basically survive, right? Here, this herpes virus and pox virus basically are producing different versions of this anti-inflammatory cytokine IL-10. So what is the purpose of anti-inflammatory cytokine? Is to suppress inflammation. If you're producing like analogous molecules to that, it is suppressing the immune system. So the virus can go undetected. So what I'm trying to say is, but with all these cases, you also learn things, because it also tells you how you can manipulate. So how you can use this knowledge to devise systems where you can, where you want to shut off inflammation. Maybe these proteins can be made in a different way without the viruses. Then you can use them for anti-inflammatory therapy, right? So everything you learn always has, you have to think how you can use it for your own good in some way or the other. Some can be used, some cannot be used. So some of the innate immune systems basically do this. I told you about this earlier, opsonization and complement activation. Here, they use the adaptive system to get rid of the organism. Opsonization, because many a times the antibody can bind to that protein also and remove the, it kind of marks the protein, bacteria for removal by the innate immune system, by the antibody, by different things. So earlier it was believed that the toll-like receptors and all these, pattern recognition receptors are expressed only by innate immune cells, because lymphocytes really don't need them because they are the adaptive immune cells. They are not going to come in direct contact with the viral bacteria and other things. But now people are realizing that lymphocytes do express toll-like receptors and other molecules. But here the function seems to be different. It is not so much to recognize the viruses, bacteria, but more it is used more to enhance their own immune response. So it's more functions as a co-stimulatory receptor, like it enhances the function, their function. It is not more for getting rid of the virus bacteria. Ultimately, it does serve that function because if their response is enhanced, they'll be more efficient in doing their function. But the function is a little different from what is there in the innate immune system. So I think some of the things I've already mentioned, that dendritic cells are like a bridge. So they bring antigens from the site of infection and present them to T cells in the lymph node. And this activates T cells, making two kinds of T cells, T helper cells and T cytotoxic cells. So this is something I will show you in the next lecture because this is something my lab also focuses on. What we try to find is how we can activate maybe the toll-like receptors or C-type lectin receptors by various components and get different kind of immune response. And this is just to show you that when the bacteria like E. coli engages TLR4 and 5, it leads to the production of IL-12. So how the dendritic cells are or macrophages are becoming activated, it also dictates the kind of cytokine they will secrete. It's not that they will get activated. So example, if they get activated by TLR3, 7 and 9, which sends nucleic acid, it will lead to production of more interferon, alpha, beta, which is type 1 interferon. If it gets activated by an E. coli bacteria which goes through TLR4, it will produce, the dendritic cell will produce much higher level of IL-12. And the cytokines which are secreted here by the innate immune cell kind of decides what kind of an adaptive immune response you will get. Will it be antibody mediated? Will it give you more Th1? Th1 in this case is interferon gamma producing cells. Will it give you more Th17 or IL-17 producing T cells, or will it give you more Th2, IL-4, IL-5, IL-13 producing Th2 cells? These are more in allergy. Similarly, some fungi and other things, when they engage receptor on dendritic cells, they produce all these anti-inflammatory cytokines like IL-10, TGF-beta, which give more of an immunosuppressive immune response. In this case, the T cell is of a different type. It's known as T regulatory cells. They will not produce any of the pro-inflammatory cytokine. They will produce IL-10, TGF-beta, which will suppress the immune response. So if you know how to activate your cell so that you can get the desired response, you can use it for therapy.
Interferon ILs as a diagnostic tools for detecting the type of infection?
So some people do believe that in many cases, people who suffer from viral infections and other things, people do look at interferon producing capacity of these, as a diagnostic tool to see that maybe this is the defect, this is where the defect lies, and can you give interferon as a therapy to these people and they will be able to fight viral infections. So there it is used. In fact, sometimes in lupus and other diseases, also toll-like receptors become a problem, and there's a lot more production of interferon. So in that cases, in fact, in lupus, it is now being used as a diagnostic where if they see high levels of type 1 interferon, they start suspecting lupus also, and then they do a whole, there's known as interferon signature genes. They do a whole array to see if those are activated. So it is being used as diagnostic. So these are all still more, they are in phase 3, phase 4, like they are going to be out in the market very soon. people have already started using them in the lab in some places, but not always can they bill. So sometimes it's more for research purposes or research labs. Okay. So innate immune system is older than adaptive, and toll-like receptors are only present in animals. But PRRs, pattern recognition receptors are present in all, even plants, and because they also need to fight infections for themselves, right? It's not only that we have infections. So this is like the basic part of the lecture, to tell you that these are the basic mechanisms which are involved. And now we will do another part where we will talk about how we use this for therapy, and where these different, how you use these knowledge of the PRRs for therapy, and what are the progress which have been made over the last 20 years or so. Because so we'll give 2 minutes and then we will start.
Madam, I have two queries interconnected with. Yes. We discussed regarding with the probiotics and prebiotics. Thing is that it is really a immunobooster, or just because of the bad bacteria get removed due to the loving bacteria. It is promising that whether the companies are promoting that it is a immunobooster.
So I'm not 100% sure it is an immunobooster. I personally think that it kind of produces a lot of good things which also act on the immune system maybe. It's right now, I would say, a gray area. It's simply people know that if you have more of this bacteria, this right now it is not even they know which bacteria. They know that this genus of bacteria is better. So that's what they will try to repopulate with, and it shows a beneficial effect. The mechanisms, I am not 100% sure that people are very clear about. Every day I hear new things which are coming.
Yeah, that's because I asked a question. Second interconnected question with this, that the naturally occurring bacteria in the guts, or somehow the fungi are also a loving one. So the new bacteria which is introduced in the body, like that of the Bacillus clausi or the Saccharomyces boulardii, these are currently right now much more flooded in the market because of the antibiotic resistibility. So they are coming with the strain, and the different companies producing the different types of the strain, and how it will compromise with our immune system, that the flooding of the different types of the strain in your body, and the immunity which is producing or responding to them, is it not the confusing for the body, that the different type of the bacteria, though they are loving, but introduced in the body?
So I personally believe that if these bacteria are not in our body, not necessarily all of them are in our body, because there are some of them are engineered also, right? So I think our body is going to make an immune response against it. But some of the bacteria are such that their immune response is not of a very high level. So that's why they go, I've seen that many of these microbiome bacteria, if you take outside and if you put with dendritic cells or even with macrophages, they don't seem to activate them. So that's why the immune system doesn't get fully activated. But I'm not 100% sure that we will not see too much of a suppression or some other kind of an immune response if we increase the number of the good or loving bacteria. Sometimes I wonder if the response, the balance is there because it is beneficial for us in some ways. But it's also a question of that if unless we perturb this balance and look at all these things, we won't know what is happening. So in all cases, you will see all these therapies which have come. We go with a very unidirectional approach. We think that we are giving this should happen, and this should result in this. But once it moves from, sometimes clinical trials and others are much more, you can focus on a number of things and do as you please. But when it comes to real world and real people, it does not behave. So many drugs fail because of this. So That's the reason I. So we'll give it a. I'll take a 5 minutes break and then I'll start the next. Okay. Oh.
Hello, hello, Mitch.
येते ना. Ah,
Hello, mic check.
On है. अब हम नहीं नहीं ठीक. हाँ चलेगा. आपको दिक्कत है? नहीं दिक्कत नहीं है. इसको हटा. अच्छा आप फिल्म कर रहे हैं? अच्छा भूल गए. अरे बाप रे. सब देख रहे हैं. वैसे ये टेक्नोलॉजी का ये एडवांटेज तो है कि एक तो काफी लोग देख लेते हैं और that's good. So काफी so I should be more careful about what I see. Okay. So far we have been talking about, all these different receptors and things, and I have mentioned some of the uses, how you can use them, but now this part is more like a tutorial. So we are already having discussions, which is very nice that people are asking, different questions, because some of it I don't even know what you ask, but I always try to answer to the best of my knowledge. Here I'm going to give you some examples how these how the defects in these PRRs are causing problems, and how they are being used for different kind of immunotherapy, or how you can potentially use them. Because ultimately one of the things which should result from basic science is it should be helpful to the mankind in some way or the other. So either it can be therapy, or that knowledge may help in something or the other. It need not always result directly in a therapy, but the knowledge should be beneficial in some way or the other to, and it's sometimes we are unable to decide at that particular point how that basic science will answer some problem which is there with something, but it's always better to keep on thinking in that direction, because I think one of the reasons we study science, as I said, is it should be helpful to us in some way or the other. Is it? I'll change it. So here it's more a recap. हट जाए गिर जाएगी अब ठीक है. So here is like a recap. So what we are recognizing is the glycans, which can be lipoglycans such as lipopolysaccharide, a component of the gram bacteria, negative bacteria, outer membrane, peptidoglycans such as bacterial muranyl dipeptide. Then this is, so these are basically different components which are glycans. This is beta-1,3 glucans from the cell wall of various fungi species. And then we can recognize proteins such as flagellin, which we have seen, nucleic acids. So all these are different components of the microbes, from the cell wall to the proteins, maybe on the cell surface, then nucleic acid, and then location. So microbial nucleic acids may be found in inside the endosome, because that's where they are found. That's where the bacteria or the fungi will be endocytosed, because that's where they will go. So the nucleic acid are found in that area. And then we also recognize the DNA, which contains a lot of unmethylated repeats, which are known as CPG islands. In fact, now people don't call CPG islands, they call it ODN, oligodinucleotides. And that's how, in fact, what people have done is they have synthesized these regions of these demethylated sequences. It's like synthesis, and then these are sold as ODNs, oligodinucleotides, and they are used, I think they have moved quite far in clinical trials for activating the immune system. So this is a slide I had made because if somebody wants to remember all four kind of different PRRs which are there and what they recognize, they don't need to go through my whole lecture. They can take this as a, it's like a summing up of most of the things, if you see, of previous slides in some ways. So here what I show is that there's CLRs or C-type lectin receptors will recognize different glycans, especially from the fungi. And here it also mentions the signaling pathway which gets activated in response to the activation of the CLRs. So in case of C-type lectin receptors, it has been shown that this enzyme SYK kinase is most important, and it leads to activation of this CARD9 MALT1 kind of a activation adapter complex. So this dectin-1 is a C-type lectin receptor. It homodimerizes with TLR2 and TLR6 also in some ways, and this recognizes the beta-1,3 glucans of the various fungi. And this, we have studied this in our lab, so I will show you some data also with it so that you can better understand how we use these receptors for various things. And here is the toll-like receptors. So they can be in plasma membrane or endosomes. And major pathways which are activated by these are MAP kinase and NF-kappa B. And similarly, you have the NOD-like receptors or the inflammasomes. So the inflammasome is basically, it is a multi-protein complex which is formed. And in that, the caspase gets activated, which basically makes this IL-1 beta, IL-18, now people are saying IL-33 also. All these cytokines are different from other cytokines in the sense that they come in a pro form. It's pro IL-18, pro IL-1 beta, and these pro forms are not active. When this inflammasome complex is formed, this caspase-1 is an enzyme which cleaves this pro form. So these are now released into the system, the IL-1 beta and IL-18. And that is why this inflammation, and these cause a lot of damage, especially when they are produced in response to danger-associated molecules, like from our self. So for this, I was mentioning uric acid. The other So what people have realized is that this inflammasome or this nod-like receptor basically gets activated in response to any kind of particulate material. So in the vaccines, you are given alum, right? So vaccines are usually formulated in a gel of aluminum hydroxide, which is alum. Alum also forms like a particle, it's supposed to have a depot kind of an effect because it's a gel. So it's supposed to cause the slow release of the antigen which is there. But this alum also activates the inflammasome because it is slightly particulate in nature. Similarly, I had already mentioned uric acid crystals. Other things is like cholesterol crystals, or basically any particulate material which comes, this asbestos particles which are the workers inhale. All these things activate inflammasome. So inflammasome does two things. This caspase, it causes release of this IL-1 beta and in some cases IL-18, which cause a lot of damage. But it also activates a killing pathway because caspase is an enzyme which is involved in killing of the cell. So ultimately, the cell will also die mostly when it produces all this IL-1 beta and other things. And the RLR are more also similar to this TLR3 and others, which recognize all the antiviral nucleic acids. So here is one of the timeline which I didn't show there, but this is, I told you that early 2000s or near about that time was when these toll-like receptors were discovered. So in 1997, Janway and Medvedov basically discovered that there is a protein which is similar to Drosophila toll, and which could activate a receptor in immune cells known as toll-like receptor 4. So with this time, I would say Janway, you can say, is the father of innate immunity in some way, because he's the one. Earlier, people never used to study innate immunity. They would think that's of no use because it doesn't even give us memory, or what it does is it just goes and activates the adaptive immune system. But with the advent of this particular discovery, the innate immune system became more and more important. I would say the last 3 decades have been focused more on innate immune system than adaptive immune system. So this Bruce Beutler, basically he used to be in University of California, San Diego. I've seen him quite a bit. So what he does is he makes all these mice, and he does this forward genetics where you introduce mutations into the mice by feeding one kind of what is it known as? Yeah. So it causes mutations, random mutations. And then he checks for, it's a lot of work. With each mutation, it leads to generation of mice. Some are not even viable. Those he doesn't worry about. The ones who are viable, then he tests. Some of them manifest, phenotype, which is clear. They're not of hair, or they may have some other problem. Those are phenotypes. In some, you don't even have a phenotype. Then he will inject them with some kind of a virus or bacteria, or see what is happening. Are they able to respond or not respond? With all this forward genetics, he found that there's a mouse strain, C3J, which basically is unable to respond to this bacterial lipopolysaccharide. So let's say that with all this hard work, but he got the Nobel Prize in 2011 for this particular. So he has not only found out TLR4 gene, he has also found a lot of mutations in mice, which are important for various signaling in various diseases and things like this. But for his discovery of this particular thing, he was awarded the Nobel Prize. Janway never got the Nobel Prize. See, Janway was Medvedov was a PhD student of Janway, or postdoc of Janway. And Janway died very early, so that's why he did really. And Nobel is not given to posthumously; it is always given to living people. So that's why he didn't get a Nobel Prize, and Beutler got lucky. Many people say that he got lucky. So this is a better slide than the last one, which was a little. So it just shows you that how many different things can be recognized by all these. toll-like receptors. These are double-stranded RNA, single-stranded RNA. These are all inside ones, and these are all on the surface. Yes, the reason these receptors are present in the cytosol is they recognize all the nucleic acids. So the virus has to go in, then only the nucleic acid will be available. So that's why these are all present in the endosome. So this is just to show you that whenever any kind of pathogen comes in, this is just showing a toll-like receptor, but you can consider any kind of receptor. It will lead to activation of host defense mechanism. Immune inflammatory genes will be activated. In case of antigen-presenting cells, you will have upregulation of antigen-presenting machinery, which is MHC, co-stimulatory molecules. Then there will be production of different kind of things like antibodies from adaptive immune cells, cytokines, chemokines from both kind of cells, adhesion molecules, and ultimate result is that there is eradication of pathogen. And so these are basically different kind of adapter molecules which are involved. So TRAb and MyD88, if you see, is common in all of them. So one of the things which you may know is that lipopolysaccharide or sepsis is usually caused because of very high reaction to lipopolysaccharide, which leads to a lot of very high level of cytokine secretion. And one of the reasons why lipopolysaccharide is so active is, if you see here, in this case of TLR4, you have 3 adapter molecules. In most cases, you have like one, this MyD88, only MyD88. In this case, you have TRAb, this MyD88, this whole complex. TRIF is also there, and TRAM is also there. So that's why the response is for TLR4 is highly amplified. It's a very, very strong immune response when there is lipopolysaccharide. And that is why sepsis is such a strong inflammatory reaction. Because multiple molecules are involved, and there are also co-receptors. So even a fraction, if you look at TLR4 expression on cells, it is very, very low level expression on cells, because even that low level is so much active that you don't need high expression. If it's too high, it's going to basically kill you. So that's one way of showing how a small, one receptor can amplify the response to such an extent using different signaling molecules at one time. So these are some, we call it experiments of the nature in some ways. So how people realized that why toll-like receptor signaling is so important was, first, there was a paper in where they found children with this IRAK4 mutation. So if I go back, so this IRAK4 is here, you see, downstream of this. So it's important for the signal to go down via this. So they found that some of the children which came to the clinic were highly susceptible to this pneumonia infection. And they were not, and then people did tests that they took out their blood and they put these toll-like receptor agonists and looked for cytokines. They could not find any cytokine secretion. So they thought they are not able to respond to toll-like receptor. And then they did a whole, mutational analysis and for these, the molecules which are there. And that's how they discovered that they have a mutation in the IRAK4. So the toll-like receptors are present, but the cells are unable to respond to them. And the children are highly susceptible to, these infections, S. pneumonia and S. aureus. So IRAK1 does the similar kind of thing. And they found that in atherosclerosis patient also, it is involved in production of IL-10. And the IRAK1 is somehow modified and it is localized in the nucleus. So it's there, but it's no longer present in the cytosol where it should combine with the adapter molecule and signal. So okay, maybe I can tell you here only. So it should be in the cytosol and combining with IRAK4 to make the signal here. Instead, it goes into the nucleus. So it is there, but it is unable to function. So then they also found children with this pneumo mutation, same problem. If you see that pneumo mutation comes here in the NF-kappa B pathway. This is pneumo. So several polymorphisms for this have been discovered. IRAK4, all these mutations are discovered. TLR4 in different mutations, different response, decreased response to inhaled LPS, and then in this case, CD14 is a co-receptor for TLR4 in some cases. So again, if this mutation is there, you have increased response to LPS because co-receptor is overfunctional. So basically, these are all present in patients. Here, this is another mutation in which NF-kappa B is not functional. And see, if the immune response is not activated, the innate immune response is not activated, it will not lead to the production of cytokines, neither will it lead to activation of the adaptive immune system. So that's why there's this defect in the memory which is shown here. Specifically, maybe these people had only looked at memory, but it is adaptive immune system needs to be activated because then only the T cells will get activated and the innate immune system will, the adaptive immune system will function. So here is a table which tells you basically how these are all the problems which are associated with the presence of these toll-like receptors. And so mostly you will see that most of the things which I have mentioned here are all toll-like receptor related. They're either their signaling molecules or their expression is resulting in all these different kind of problems. Most of the time, if you see, these are leading to some kind of an high level of infections, right? Yeah, this is in tuberculosis, the MAL receptor, or it is leading to some kind of an autoimmune disease. SLE is basically systemic lupus erythematosus, so a lupus disease. Where what I had said is that it recognizes, the toll-like receptor are supposed to recognize viral and bacterial nucleic acids, but in this case, SLE, they are start to recognize their own nucleic acids for some reason or the other as foreign. So similar to what they will do in virus and bacterial nucleic acid, they produce a lot of interferon. And that's why you see this mutation is an interferon response factor 5. And that's why this needs to increase susceptibility to SLE. IRAK4 is less, it's not associated with, if you see, SLE in some ways. Only IRF5 is mostly associated, and you will also see, yes, somewhere here. Mostly it is the toll-like receptor 7, 9, which are very, very highly involved in SLE pathogenesis. So in this whole class of immunodeficient patients who suffer from a lot of these infections are known as primary immunodeficiencies or PIDs. And in most of these cases, these are from birth. Mostly there are mutations in different regions of, either TLR4 or signaling molecules. And the only way you can either treat them is either you have some kind of a gene therapy, or if you're able to provide a stem cell transplant in some ways so that they are no longer producing that defective cell. Because many of these children will not survive for long because the amount of infections they face is so much, recurrent infections all the time. And pneumonia, if it occurs many, many times, it's always a major, major problem. So because of this, there's been a lot of, as I said, interest. But this is just to show you that all these, it's given in this table also, I've mentioned earlier also, all these are involved in different diseases. These are all genetic defects that are there, and don't worry about inheritance, and these are all different kind of infections which come. And what are the different TLRs which are involved in this problems. This you can choose to study whoever is interested in what, because the list is growing every day. So this is just an example how the toll-like receptors are also involved in something which is very unrelated, which is a hyper IgE syndrome. IgE is a kind of antibody type which is produced mostly in allergies. And if it is too much of an IgE, then it is going to lead to a lot of hypersensitive reactions. So what they realized is that there is a dominant negative mutation in STAT3, basically here, and even in DOC8, and this leads to somehow high levels of IgE being produced. So this was very considered very unique, because so far most of the other things have always led to some kind of an high infections and other things. Never a change in an antibody isotype suddenly leading to this kind of a mutation in an innate immune system cell, leading to high production of one kind of an isotype of an antibody. So that's why it was considered very interesting by most people. So apart from the infections, though patients which I have mentioned have a genetic defect, but TLRs seem to be also involved in a number of diseases where, because they recognize these molecular patterns, right? So these molecular patterns can also be produced by damaged cells, because they also release sometimes some proteins, some things which are recognized by these toll-like receptors. TLR4 is a major example. It seems to recognize many of our extracellular matrix components like hyaluronic acid, or uh, sometimes a mixture of this collagenase and other things are also recognized by TLR4. And in case of rheumatoid arthritis and others, these components are supposed to cause inflammation of the joints. So if they are released in the system, and so these are these are known as sterile inflammation, because inflammation is not there because of some kind of an infection or other things. It's because of a natural component which is our own part of our body. So sterile inflammation is considered major problem in most diseases. Here is an example of rheumatoid arthritis, but it has been shown in the joints that TLR4 and TLR2 are upregulated in the joints of rheumatoid arthritis patients. Atherosclerosis is another example. Sepsis, I already mentioned, lipopolysaccharide of the bacteria are major problem in sepsis. So, and the list is growing every day. This is just to show you, like you see, it does cause protective immunity, but there's septic shock, asthma also, TLR activation is involved, COPD, ARDS. So it has a number of different problems which are associated. So these are all mechanisms. These are all activation in the epithelium, in the innate immune system, leading to activation of T cells. But these are all the problems which are associated with the toll-like receptor not functioning properly, or overactivation, or not, or a mutation. So here is an example. You see, these are endogenous and exogenous ligand, right? So these are all things which are produced by our own body, hyaluronic acid. So it is, I thought it goes by TLR4. So fragments go by TLR4. Some fragments are also recognized. This is high mobility group box protein 1. This is produced mostly by cells when they die. So if they die not a natural death, so when cells die, they die by apoptosis. Apoptosis, what happens to the outside, but nucleic acid and other components are released, and that's when the immune system is able to see them. If they die by like apoptosis, since they die in such a way that nothing is exposed outside, our immune system doesn't see our own nucleic acids and proteins. It's when you have cells die because of maybe toxicity, or so necrosis could be simply because and the apoptotic cells are cleared up by phagocytes. So macrophages, neutrophils, apart from clearing up bacteria and fungi, or bacteria, viruses, whatever comes in, they also have the other function. When cells die by apoptosis, these are also removed by macrophages and neutrophils. And if for some reason they are unable to remove these apoptotic cells, then they will burst open. So, and in lupus and in many other diseases, in fact, a number of autoimmune disease, it has been shown that this clearance process by macrophages and neutrophils is defective. So that's when the cells have burst open, and it has led to the innate immune system becoming activated and able to get hold of these components which can activate them. Danger signals are released. So this is, as I said, there are so many things. If you see oxidized LDL, fibronectin, heparin sulfate, or HSPs, all these are recognized by these toll-like receptors. Apart from, the normal, these are all bacterial cell wall components which are recognized. So that's why these are the problems, because in many of the diseases, this recognition of these cause the problem. So these DAMs, as I said, danger-associated molecules, are only released when there is a tissue injury or cell damage, and the problem with danger-associated molecules is once there is an injury, and if it is not able to resolve, this danger-associated molecule is going to be produced continuously, and continuously the innate immune system is going to get activated, which will lead to more inflammation, more tissue damage. Again, the whole cycle keeps on continuing. So that's why this activation of the innate immune system by DAMs is a major problem in autoimmune diseases or in other diseases. When it reaches a certain threshold, it will lead to some disease. This is just to show that in case of MIDI, how people realize that TLRs are involved in atherosclerosis is they made mice which were deficient in MID88 and ApoE. ApoE is a gene which seems to be very involved in a number of, if it is overexpressed, it just seems to be responsible for susceptibility to a number of diseases. Atherosclerosis is one. Alzheimer's is another one, where you have it depends on which allele of ApoE is present. Like allele 4, if it is present, there is very high chances that Alzheimer will be. So they realized that when they made these mice which are deficient in this MID88 and also ApoE, even in the MID88, they realized that even if you give them cholesterol-rich diet, which should cause atherosclerosis, these mice are resistant to atherosclerosis. So most of the discoveries have, as I said, come with by the use of these knockout mice which have been made, so that's why people consider Akira also like a father of TLR, because he made most of the mice which are involved in this toll-like receptor discovery pathway. So this also, I have mentioned this part. So the confirmation came by getting the mice which are resistant to TLR4, and in these mice, the LPS-induced shock is not there. And again, mice lacking TLR4 are also more sensitive to infection with gram-negative bacteria. So if you don't have TLR4 also, it is a problem. Then you're going to be more susceptible to the E. coli and other infections, because all these toll-like receptors are also involved in protection. So here is an example of the autoimmune which I was mentioning, that the TLR7 and 9, they are supposed to recognize the viral SS single-stranded RNA and the CpG DNA. Basically, TLR7 recognizes the RNA part of it, and TLR9 recognizes the CpG DNA, which is more a bacterial DNA. And this, so this is like detailed explanation of how the activation occurs, but don't worry about it. Basically, once the virus has reached the cytoplasm, it goes into the phagosome. The viral nucleic acids come into the endosome, and here the TLR7 is there, which gets activated, and it leads to production of type 1 interferons. In case of damaged cells, this particular complex is released. This HMG1, there's a protein known as high mobility group box protein 1. It forms a complex with DNA. when the cell breaks open, this complex is accessible now to be taken up by other phagocytes, where it goes inside and does the same thing as a virus DNA will do. So that causes problems. Similarly, this LL37 is an antimicrobial peptide, but it also binds to DNA, and it is used for taking in the DNA, our normal DNA. So these are ways how our normal DNA reaches these TLRs to make them activated. And these pathways seem to be overactivated in the diseases like lupus is definitely one. Even in Sjogren's syndrome or rheumatoid arthritis, you see this danger activation of these TLR7, 9. Mostly 7 seems to be more involved in lupus. Okay, there is also one theory in general that these autoimmune diseases like lupus, rheumatoid arthritis seem to be more prevalent in females. In fact, it is a 9-to-1 ratio in case of lupus and other reasons. And generally, it is. It seems the evolution is such that females are less susceptible to viral infections. Our TLR7 and 9 or production of interferon pathways seem to be more robust. So the downside of it is that we also are more susceptible to autoimmune diseases because our response to nucleic acid of our own is also more robust because of this. So there's also a lot of research going on in this dimorphism, these receptor expression or response. So earlier, mostly people thought that it is mostly the TLR7 which is involved in this interferonopathies or interferon-related diseases, as they call it. But now they're finding that a lot of other receptors are also involved. RIG1, MDA5, these are also basically intracellular receptors which are present in the same place where the these TLR7 and 9 are present. But the only thing they do is they recognize maybe different portions of the nucleic acid, like phosphate group of this. But ultimately, they are able to recognize nucleic acid of foreign and endogenous origin. So they are also involved in autoimmune diseases. Similar to this, the nod-like receptors, which I mentioned, inflammasomes, some of the inflammasomes are also able to recognize DNAs and other things. That's why the interest in these have become more and more over the last 5-6 years. So this is just to show you that in case of polymorphisms in RIG1 or MDA5, so many diseases have occurred. This particular disease is Singleton-Merten syndrome, AGS, and then SLE, type 1 diabetes, Graves' disease. In all these cases, there is some involvement of some mutation, something or the other, which is leading to this particular, these diseases, AGS, atypical. So what I'm saying is, as long as these receptors function normally and recognize foreign things, it's fine, they provide protection. But if there's any mutation, any problems, it will lead to diseases. So it's a lot of different things, but if you want to read it, I have given it here because that way whoever is more interested can go ahead and read. But the bottom line is that RNA or DNA can be recognized and lead to production of interferons, both foreign. The RNA and DNA can be of both foreign origin or our own origin. Normally, our own DNA, RNA have certain modifications which make it more difficult for them to be recognized. The other thing is they are also less accessible to these receptors. But if for any reason they become accessible, then they do. These receptors can respond and lead to production of interferons, which cause a lot of problems. Interferons are, they're supposed to fight viral infections, but if there's no virus to fight, then they result in a lot of problems. Because if you see, when you have a flu infection, you do feel very tired, right? So the same thing will happen. It's basically trying to fight a flu infection constantly because this stimulus is there constantly. So it's going to lead to a lot of damage. So which ultimately results in some kind of a pathology. So a lot of molecules have been discovered over the last few years, which all recognize basically DNA. You know, I've been talked about RIG1, MDF5, but there are so many receptors. Now I'm here going to talk about this DAI. This is ZBP1. This also recognizes this DNA. Sting mutation, you may have heard. Sting is another I don't even remember the full format. It recognizes also DNA. So a lot of interest has been generated in which are the molecules inside our cell which basically recognize DNA. So in fact, over the last 10 years, if you will see a lot of papers are there in high profile journals which every day come out identifying one receptor or the other and it's signaling molecules, CGAS, STING, AIM2, and then the different kind of inflammasomes which recognize different structures. So this seems to be right now a major hot area of study. So I was talking about the nod-like receptors. So if you can see here, so these are different kind of nod-like receptors, NLRP1, B, NLRP. So this is the most studied one, is what I'll say, NLRP3. And this is what recognizes most of the crystals which are formed, like endogenous dams, what it is saying, the crystals, and it leads to cell death and production of IL-1 beta ultimately. These recognize salmonella and these are recognized DNA viruses. So major thing is that they are involved in recognizing different pathogens. And the major thing which the nod-like receptors do is production of IL-1 beta, IL-18, which seems to be very important. But the inflammasomes have become more important from another point of view. Over the years, what has come out is that they seem to be involved in a number of diseases. All these diseases seem to have a major involvement of this over inflammasome associated diseases. In all these cases, there are different kind of things which are activating the inflammasome. They are danger associated molecules. In case of heart, metabolism, type 2 diabetes. In cancer, this is suppressed to a certain extent. Intestine, even in the inflammatory bowel disease, this seems to be chronic acute inflammation. So in most of the diseases, it seems to be overactivated. And as I said, the major thing which happens with this inflammasome complex is pro-IL-1 beta cleaves this IL-1 beta. And this caspase 1 here, it gets activated. It causes killing of the cell, which is here in more detail if you want to see. That this is cholesterol crystals, or you can take oxidized LDL, free fatty acids, which are recognized by TLR2. But here, ultimately, as I said, in all these cases, this whole complex assembles and there is production of IL-1 beta. And beta cells also. So I think we need not worry about, unless you are studying this in great detail, that how it is formed and what it is formed. This is just to give you an example that in case of type 2 diabetes, all these things are present, and this and the so inflammasome seems to be more active in macrophages and somehow or the other. It is not that it is restricted to macrophages, but so far it forms most easily and most easily detectable in macrophages. And most of the studies have also been done in macrophages because of this. I know that in dendritic cells also it forms, in neutrophils also, now people are showing in epithelial cells also it is formed. But most of the studies so far have focused somehow or the other in macrophages. So that's why whenever you will see about toll-like receptor, this nod-like receptor inflammasome, you will see that most of the the studies have been done in monocytes. So all these things can activate the inflammasome. This is, and as I said, the ultimate production is these two. But how it leads to insulin resistance and organ dysfunction is not still very clear. This is a pyrogen. It causes a lot of damage, but what it does is still something. So here is, so these are from different journals, different papers, where people have studied these, how the inflammasome is getting activated. So because of this, there's a lot of interest in developing therapies which can prevent this inflammasome activation. Because if it is involved in so many diseases like atherosclerosis, type 2 diabetes, then if you can find something which is, in fact, any kind of a metabolic syndrome seems to involve in activation of inflammasome. So there is a lot of interest in developing inhibitors which can suppress this inflammasome activation, because this would lead to, cure of these diseases, or at least some kind of a mitigation, less inflammation in case of these. And in fact, people have used antibodies against this inflammasome and shown that it seems to be beneficial, which I will maybe come a little later. Again, tables of NLRP3 associated diseases and what kind of problems is there. So these are, okay, maybe I can set the next slide also. No. So these are all inhibitors that are in various stages, which people are trying. These are all developed to inhibit inflammasome activation, and mostly inflammasome, NLRP3 based inflammasome, because this is the one most studied so far. And these are the references, which are there. I think this is from this paper, so you can go back and look at it if you wish, in all cases. So, but it's a very highly active area of inhibition. And the reason also people are very much interested in this is, if you see the diseases I mentioned, they affect a vast population. So if you come up with any kind of inhibitor, it is going to be, the companies will make a lot of money. So there's a lot of investment in it as such. So those were the inflammasome based receptors and their problems. So now we come to C-type lectin receptors. So here, these are all different kind of, you can see, they also recognize a lot of matrix proteins. Okay, DC sign, Minkle, but many of them recognize a lot of fungi. So this, I would say, the least studied of the PRRs, because I had mentioned this earlier also, these, the C-type lectin receptors don't get activated to a very high extent, and they don't seem to be involving the normal pathways which are mostly associated with inflammasome or TLR, toll-like receptors or RIG-like receptors. So that's why I feel that they have lagged behind to a certain extent. They are used more as markers for cells, there is these receptors, or few have been known for their uses. So an example is this C-type lectin receptor Minkle, which is present on macrophages. So it recognizes cholesterol sulfate, but ultimately it also leads to activation of inflammasome, and which causes damage. But more importantly, I think, yes, this is the one I was saying. The reason the C-type lectin receptors are known are, especially the first one, the very early one which was discovered was DC sign, and it is present on dendritic cells, and it basically binds to the HIV virus, and transports it. So this is the molecule DC sign. This is a mannose receptor, which is also one kind of a C-type lectin receptor. These can bind the HIV-1. So on one hand, it is good because it activates dendritic cells and it can try to kill the virus. But the major problem which happens is that dendritic cells, when they become activated, or their major function is they have to go to the lymph nodes to activate T cells. And if you know the HIV virus, what it does is it infects your CD4 cells and kills them. So in this case, the binding of the virus to DC sign, though it activates DC to a certain extent, not too much, DCs basically act as carriers of the virus because they are taking them to the area of lymph nodes where the T cells are. So now the virus can infect the T cells and get rid of the T cells in our body. And that is one of the reasons HIV virus survives for so long, because it kills all the T cells. There is no immune response against the virus. So, and the problem is, how do you improve that immune response? Because the T cells are going. So that's one of the reasons I think at least the immune boosting therapies for HIV are not so successful. It's the antiretroviral therapy which seems to work a little better. But most of the patients for HIV die more because the immune system is compromised. So they get a lot of other infections. So now, so far we have been talking about how all these different receptors are involved in diseases because of either mutations or because they're overactive. Now we are coming to how we can use them for therapy. Okay, if you find out the mutations, maybe in some ways you can correct, but I think it's still gene therapy is not so easy to do. But these are the part of the talk. So far the toll-like receptors have been used for a number of different things, drugs. One is vaccine adjuvants, and this I have mentioned 2-3 times, how to modulate the adaptive immune response to the way we want it. And this is something which I think is still quite a bit focused on, because every day I see new things which are coming out. So in this case, in case of sepsis, there are many TLRs which are involved. Major is the TLR4. So if you can develop an antagonist of the TLR4, something against MyD88 will also work, right? Because that's downstream. That was an adapter protein which was downstream of most TLRs. So if you can make a blocking agent against that, so it will inhibit TLR signaling. So it will work against sepsis. It's not that you have to block it all the time. You have to only give it to the patients. So short-term blocking is fine. In asthma, again, you have toll-like receptors, involvement of toll-like receptors 249. So some are overactive, some are underactive. So you can try agonists, antagonists. So that now, so here is an example where in case of asthma and allergy, most of the time the immune response is kind of a Th2 immune response. Th2 immune response means that T cells will be producing IL-4, IL-5, which is basically an allergic kind of an immune response. So you want that the cells stop secreting that and start making the opposite, basically interferon gamma in this case, or Th1. So this is using these agonists, you're trying to modulate the immune response. In cancer, a number of these toll-like receptor agonists have been used because in cancer, what is happening? You are having a suppression of the immune response. So you want to use something which is going to enhance the immune response in some way or the other. So this has been tried for decades, that people have given small amounts of bacteria or something to activate non-specifically the immune system. And as a bystander effect, they think that the immune system cells in the cancer will also become activated.
There is a tumor in the body. So can we put an inflammatory product directly to the tumor, like a bomb? You put an inflammatory product directly into the tumor, So the immune cells will be recruited and it will
So
shrink, right?
this is an approach which has been tried. It has worked successfully in a few things, but not all the things. See, when you're putting it directly in the tumor, the problem is also that the tumor itself is secreting a number of factors which suppress the immune system cells. So even if you're putting off the bacteria attracts the immune cells, it has to be in sufficient numbers to break, because tumor and the microenvironment both are producing so many factors. Every day there's one new discovery which is actively suppressing the immune system. So even if the cells are coming there, they are also going to be suppressed, right? So that's why not in all cases it seems to be working. In fact, it seems to sometimes work when you do it in the vicinity of the tumor than the tumor itself, because then you are slightly away from the tumor suppressive factors. In that case, the immune system is getting activated, and those cells may go there and activate, result in breaking of tolerance in the cancer. So one place where it has been very successful has been in, somehow nobody still knows why, bladder cancer. In that case, BCG, which is a vaccine for tuberculosis, is injected into the bladder, and it seems to somehow break the tolerance, and it has shown to be very, very effective against bladder cancer. It's the only way people think is that it breaks tolerance, but why only that, nobody knows. It doesn't seem to work in other cases. Either the environment is contained in some way or whatever. Then some people are trying also this what they call neurotropism, in the sense they're using this polio virus modified in some ways to activate brain immune system, because it's a natural infection by a polio virus, right? And it will activate, and they think that because of this activation, the brain tumor, glioblastoma or some, should the immune system is going to attack and maybe shrink the tumor. This is, I've just read of one or two papers, and some people think that it's showing some effect, but these are bystander effects. It's not directly killing the tumor, but basically you're trying to activate the immune system, so now that it goes and kills the tumor. These are things which are trying, but they don't always seem to work. Only in like some cases suddenly they work, in some cases they don't work. So either we need to understand a little bit more that there is a certain class of tumors or certain type of tumors where it will show an effect, which I think more research needs to be done.
Why, ma'am, our own cells are transforming into cancer cell and it is acting against us only?
No, that is the problem, because it's our own cells, and immune system is designed in such a way that it should not it should not kill our own cells, right? So that is why the tumor grows, because it is not considered foreign unless it is showing something on the surface. Then other cells will, natural killer cells will kill it. If it is not showing any change on the surface, or it's not looking any different from our normal cell, then it is a self cell. It's our own cell. So it's one of the reasons why the immune system does not attack the cancer in the beginning.
But tumor cells are carrying so much mutation burden, right?
So see, the thing is, tumor mutations not always result in any kind of cell surface changes. So for immune system to recognize, it has to see something foreign, right? The cell is exactly like our cell. Unless it is expressing more of certain proteins, or if it's showing certain changes which are visible to the immune system, it is not going to recognize.
But how come then that last year they got Nobel Prize, no ma'am? CTLA4 antibody, PDL1.
PDL1, CTLA4, right?
How it works?
So that works because that is what it does. It breaks the immune tolerance. It doesn't recognize any tumor cells or anything.
Okay.
It basically activates your immune. It acts on the brake. So PDL1 and CTLA4 are all brakes of the immune system, right? And tumor uses these brakes to suppress immune system. That's what I will show you in the immunotherapy lecture. So the therapy is such that this brake which is put by immune system, they put an antibody to remove this brake. So now the immune system becomes activated. And the reason they got the Nobel Prize is this therapy, this kind of a therapy is effective against all tumors. It is not restricted to any tumor, because this is a universal phenomenon which is happening in most tumors. So that is why they got the Nobel Prize, that it is, and in many cases, see, the I think the drug which got approved is Keytruda, PDL1, PD1 antibody. I think it has been now 3 to 4 years that it has been used in patients. So far it has shown very good promise in the cancers it has been approved of. Sometimes the survival is, patients have survived.
But it
So
may
now-
work other way around, no? It may overactivate T cells and.
So there are problems
autoimmunity.
associated with it also. It's not, they are following the patients, right? So nothing ever comes without a cost. So a lot of other problems are being seen, but the problems, since they're aware of these problems, they are keeping an eye on it, and the treatment begins side by side. It's better than having the cancer, let's say. It's not that it doesn't, it leads to some autoimmune problems, but if you are aware of them, you can treat them from the beginning. So they don't cause problems. They are on a lookout for this, let's say. The patients are monitored continuously for this. Still, I would say that it's only 3 to 4 years that a lot of patients have received this. That's why there's a lot of very follow-up which is going on, because they want to see long-term. This is only 3 to 4 years, right? Maybe in the clinical trials, the ones who are there have survived for longer, but still, long-term use of these things, will they result in any problems or all, which we will come to know a little later. So far, they look very, very promising. Okay, so this is an old slide, only from 2010, but because I couldn't find any new ones like this, I think now people just assume that many of these have moved forward or whatever it is. But this is just to show how many companies are involved in making different kind of compounds that target the toll-like receptors and used for many different diseases and targeting many different kind of toll-like receptors. So if you see one of the things which is most targeted is the CPG, drug class is CPG, because making oligonucleotides is much easier, right? We do it all the time to make even primers for that matter. Everything is what linkage of nucleotides. So that's why this drug is most easily made. So different kinds of it are being tried, different combinations, things. Mostly you will see many are nucleic acid based, because those are most easy to make. So this is an example of a cream which was already in the market. It is known as Immunomod. So what they realized is this Immunomod works in keratitis and other things so it's a topical cream. But this was realized later that how it works is it really activates the TLR7 pathway, and it activates results in the production of interferon, and it seems to be beneficial in this condition. So I just gave you an example that how things like this can be used for treatment. So this is a slide which tells you that once there are different, these PAMPs, different receptors on dendritic cells. Here I have taken an example of dendritic cell, but you can macrophages. I would not say that you can maybe you can put B cells, but mostly macrophages. And when any kind of a, PAMP comes in, this is any kind of microbe. This is another microbe, PAMP2, which is activating another receptor. These dendritic cells will become activated and they are secreting. Here, this is something we've done in our postdoc work, so I'm just showing you an example. This is E. coli receptor, this is E. coli LPS, and this is P. gingivalis LPS. And it is engaging one TLR, this is engaging another TLR. Here in this case, we are seeing production of IL-12 and different other cytokines. Here in this case, we see more production of IL-10. It doesn't produce IL-12. So then you get more of a Th2 kind of an immune response because it's not producing IL-12. And here you get more of a Th1 kind of an immune response. So what I'm trying to say is that how the dendritic cell or the macrophage is becoming activated by the microbe, it results in a pattern of cytokine secretion. That pattern will decide what kind of a T helper cell response you will get. So here is an example, but I'll go further, more deeper into this example. This is just to show how you can use them for vaccine and therapeutic, because you can get this, the Th1 response is important for protection in intercellular pathogens. So if you make the cells Th1, you can use them for this protection from intercellular pathogens. Similarly, for Th2, it is more for extracellular pathogens, because in case of worms and other things, parasites, this response is important. Then Th17 is very high in autoimmune diseases, but it is very important for fighting fungal infections and pneumonia infections. So this response should be high. And this is another kind of T helper cells, T follicular helper cells, which activate B cells. And these are some movies which will show you this. And don't worry about all these different TLRs, which are, as I said, this is something for people who are very much interested in it. So I've provided this information that these are all different kind of things which are in different stages. So you can see how much work is going on. So now we come to a little bit of a different things. Mostly I've been talking about how you can modulate the immune response. So this is an example of that. So this is a picture of dendritic cell with T cell. So you can see the difference in size. This is an activated dendritic cell because you have so many of these dendrites which are coming out. So DCs, as I mentioned, are located throughout the body. They capture and process antigens, and they express two different kind of MHC molecules. One is MHC1 and MHC2. So when the antigen is presented in context of MHC2, you get T helper cell activation or CD4 T cell activation. When the antigen is presented in the context of MHC1, the cytotoxic T cells get activated. These are all different classes of dendritic cells, but I think that is more detailed, but maybe these two are the major subsets, you can say, in the blood or even in different organs. And these two can be subdivided now. Every day new subdivisions are coming, you know, this marker and that marker. But I would say that there are two kinds, myeloid dendritic cells and plasmacytoid dendritic cells. The only difference is these are also, they look like B plasma cells. That's why they are known as plasmacytoid dendritic cells. They are lymphoid in origin. And these produce very high levels of interferon in response to viruses. In many cases, it has been seen that if these DCs are lacking, people are more susceptible to viral infections. So that's why this name, maybe PDCs, you can remember if you wish. So why I have always been interested in dendritic cells from my postdoc days. So why I think is that dendritic cells, if you take an orchestra, dendritic cells are like the conductors, and they basically tell all the other cells what to do. So how they are going to get activated, they will give signals to other cells and tell what to do. So that's why if you can manipulate how they are responding to certain things, you can basically manipulate all the other cells. So, okay, so this is basically DCs. If they are producing IL-12 and some other things, then the T cells will make interferon gamma, TNF alpha, they'll be TH1. If DCs are producing more of IL-4, IL-33, then the cells will be more TH2. If the DCs are producing more TGF-beta, IL-10, we will see more of T regulatory response. Similarly, if they are producing more of IL-23, IL-6, we will see more of a Th17 kind of an immune response. So, as I have mentioned so far, we'll go now into an example instead of keeping on mentioning the same thing. So this is an example. So I mentioned that the dendritic cells in the blood are very small, very less. You have maybe 1%. So it becomes very difficult to study dendritic cells, because in 100 ml of blood, if you purify, first of all, from 100 ml of blood to purify it is very expensive to purify. You get about maybe less than a million dendritic cells. So it is very, very hard to study. So dendritic cells were discovered by Ralph Steinman, and he also got a Nobel Prize for that. He only realized that if you take monocytes from the blood and if you culture them in the presence of these two cytokines, GM-CSF and IL-4, by day 6, they form dendritic cells. And these are known as monocyte-derived dendritic cells, because now people are saying that they are slightly different from blood dendritic cells, but for many purposes, they serve as good models to study them. And most people in the lab use this method to make dendritic cells. And we also do the same. Especially in humans, it becomes very, very difficult to study dendritic cells. In mice, people can kill many mice and accumulate them from spleen and other things. They don't need to depend on the blood. So basically, when a PAM triggers immune response, there's cellular signaling, there's immune response, and this is, I've mentioned enough about it. So, and these are some of the papers we had published earlier, all based on this kind of a thing. So, and this is a summary of this, that when we had activated dendritic cells with TLR4, we get this activation of, we also looked at signaling molecules, and what we found was we get more of the MAPK activation, which leads to the production of IL-12 by the dendritic cells. And when you culture T cells with these kind of dendritic cells which have been activated with TLR4, you get more of a Th1 or interferon gamma producing T cells. And if you activate them with another receptor, which is TLR2, then you get more of a Th2 response. And if you use something against which activates only the C-type lectin receptor dectin, it leads to production of this kind of cytokines, IL-23, IL-1 beta, and you get more of a Th17 response. Similarly, if you engage both these two receptors, it is more a tolerogenic response. So here I am showing that this one cell, depending on what kind of a stimuli you are using, you are getting a different kind of a Th response. And this is, just to give you an example, here is, we have taken the dendritic cells we have made earlier, and we are looking at activation molecules by flow cytometry and supernatant once we have stimulated them for 48 hours. And the stimuli we have used are lipopolysaccharide, which is from gram-negative bacteria or E. coli, and it binds to TLR4. The second stimuli is zymosan, which is a yeast cell wall preparation. It signals via two receptors, TLR2 and dectin-1. And there is a third component. This is beta-1,3 glucan, which signals only via dectin-1. So this zymosan goes via TLR2 and dectin-1, while curdlan goes only via dectin-1. And the reason we chose this curdlan was fungi, mushrooms also are fungi. And it has been shown by a number of, mushrooms are used since centuries to activate the immune system, and especially in cancers and other things, they seem to be very effective. And curdlans are these beta-1,3 glucans are found in many mushrooms. The composition may be different of the beta-1,3 glucans, like the beta-1,3 chain may be different than this, but it's a common component, and this seems to be effective against cancer. So we were interested in knowing what kind of an immune response curdlan gives, which can explain why you are seeing that it seems to be a little effective against, it's a immunostimulant and it seems to be effective against cancer. So these are dendritic cells which have been activated with LPS, the zymosan which goes via dectin-1 and TLR2, and this only via dectin-1. So if you look at this particular cytokine, this is IL-12, LPS is giving so much of it. But in response to these two, these two are giving much less, right? So if you look at the other cytokine, which is IL-23, here now this one is giving you very high. So it's the same cell. The only thing we are doing is activate with them with different ligands, which are activating different receptors. Now LPS is giving much less of this. Similarly, if you look at IL-1 beta, again, Curdlan is giving much more of it. And the reason we think Curdlan is giving much more of it, because it is also a colloid, so somewhat particulate in that matter. So that's why I think we are activating also the inflammasome when we are giving Curdlan, that we are seeing more of more of IL-1 beta. Oh, sorry. But if you look at IL-10, it's the zymosan which seems to give very high levels of IL-10. So when you put these dendritic cells now with T cells to see what kind of T cells they will prime, because based on the cytokine pattern, we had predicted that they will give different kind, the T cells will be of different kinds which will be produced, right? So now we activated the DCs, now you put them with T cells, because that's one of the functions of dendritic cells, to activate T cells. But the way they are activating T cells is always going to be the same. But the kind of T cells which are resulting from this activation are the ones which are different. And that's what we are trying to show here. That in the case of LPS, we saw very high levels of IL-12. And here we see much higher level of interferon gamma production. It's directly proportional in a way. Sorry, one second. So you're seeing high levels of IL-12 here, and then followed by Curdlan. If you look at, interferon gamma, maximum in case of where the most IL-12 is there, followed by Curdlan, right? The other thing which was very high in Curdlan was IL-23 and IL-1 beta. And those are the cytokines which are supposed to give you T cells which produce very high levels of IL-17. So you can see this is what you are seeing here. Now if we go back, IL-1 beta is also being produced in case of zymosan. So both IL-1 beta and IL-23 are supposed to induce IL-17 producing T cells. So that's why you are seeing this kind of a gradation. These are producing also IL-17. This is the maximum, because here you saw maximum production of IL-23. And zymosan, it was producing high levels of IL-10 also. It is here also the T cells which are primed with it are producing high levels of IL-10. So what it is simply showing us that Curdlan is giving more of a Th1, Th17 kind of an immune response, and very low levels of IL-10 compared to. So it's a very highly inflammatory immune response which the Curdlan is inducing. And this could be one of the reasons why it is so effective in, it works so well as an immunomodulator in case of cancers. See, the other major thing which is important in cancer is that it should also generate T cells which are cytotoxic, which should be able to kill the tumor. T helper cells are important, but T helper cells will produce mostly cytokines, which are supposed to activate other cells. So here we basically took the dendritic cells and now put them with CD8 cells, which are the cytotoxic kind of T cells. And if you can see, mostly the most levels of granzyme are produced by Curdlan. Even the perforin expression is much higher on Curdlan activated. The DCs which are cultured with, DCs activated with Curdlan which are cultured with CD8 T cells. Zymosan is also effective because yeast has also been shown to be effective in killing tumor cells. And the difference between Curdlan and zymosan is that Curdlan utilizes only the dectin-1 receptor. Zymosan utilizes both dectin-1 and TLR2. That is the only difference. And the agonist, therefore, is different. So it seems to me that when dectin-1 is involved, it's giving you an immune response which is higher in Th17, which induces high levels of cytotoxic T cells, which may be effective against cancer. So if you look for agonist, yeah. So I haven't tried it in the xenograft mouse model because other people had tried this. They have shown in Curdlan that it does seem to shrink the tumor to a certain extent, but I haven't still seen so much of it in moving forward, like in patients and other things. I don't know why, because Curdlan to a certain extent is an easy molecule to move forward because it's used as, it's a colloid, so somehow it is used in and other things as gels. So it is non-toxic in that way, because if it's used in a food substance and it's used for a long time, then, but I think one of the reasons is everyone focuses on a different kind of mushroom. Some say this mushroom is better, especially this is more in the Japanese and Chinese community and Korean community, and each one has their own mushroom which they think is better, and the beta-1,3 glucan in all of them are slightly different. So that is one of the reasons I think. But I do know that it is being sold as immune supplement. I have seen this in the shelves in the market that they sell it as an anti-cancer, because then you don't have to do so much of validating. If you're selling it as a supplement, people buy it. A lot of immunomodulants are sold as supplements because then you don't really need to worry about anything. You are not liable for any kind of problems which occur with it. You don't have to do any testing, but you're still able to make money. So that's, and here we also use that CD8 T cells which are generated here, because sometimes people say that, okay, you have this granzyme expression and perforin expression, but does it really result in cell killing? So this is just to show that we also look, this is a cell killing, this is tumor cell to CD8 cell ratio. And this essay I can describe, and this has been published long time back in PLOS 1 or whatever it is. But if you see the Curdlan specific T cells are best at killing. The percent lysis of target cells is much higher. I think we used PC3 prostate cancer tumor cells for this. So it does kill in vitro. In vivo people have shown, in mice definitely. So this is another example. Here now I am not activating with any kind of toll-like receptors. This is just a molecule which is present in our own body, PDGF, which is platelet-derived growth factor. And this was done in collaboration with, in fact, this resulted from a collaboration with somebody else. Earlier there was a question that stem cells, why they don't, if they are defective in aged subjects. They are not defective, as I mentioned. So in that, the other group had shown that the level of TGF beta and PDGF is very high in aged subjects, and that seems to act on stem cells to make them not functional. So since I don't work on stem cells, I wanted to see what PDGF does on immune system, and I work mostly with dendritic cells, so I had put them, since the system is already there in the in our lab, we just put PDGF on dendritic cells and saw what is happening. So what we found was that if you put PDGF in dendritic cells, it induces high levels of this IL-10, which is an anti-inflammatory cytokine. Okay, and when you put them with these dendritic cells with T cells, these T cells also produce high level of IL-10. Low levels of the interferon is reduced compared to controls. IL-10 is high, as I showed. This is control, and this is, we don't see so much of a dose response, but basically there is a decrease. It's the immune system seems to be suppressed. This is proliferation. Proliferation is also lower, and when you look at the kind of T cells which are being made, there are high levels of IL-10, and they are making this FOXP3 expressing T cells. So FOXP3 is a transcription factor which is only present in T regulatory cells. So IL-10 can be produced by so many cells. Okay, even dendritic cells are making IL-10. So some people have this problem that IL-10 can be coming from this remains or that remains. So if you want to say that a cell is T regulatory, you have to show this transcription factor. And this is what is shown here, that these cells, these dendritic cells which are exposed to platelet-derived growth factor, when they are cultured with T cells, they are making high level of FOXP3 expressing T cells, which are T regulatory. So what you are getting is, when in the presence of PDGF, you are able to generate T cells which are highly suppressive. Now the question is, can you use this for therapy? So next, what we wanted to show is what is PDGF doing to the dendritic cells? Why is it resulting in this Treg, okay, IL-10 secretion and everything? So what we found is when you culture this PDGF with dendritic cells, it leads to the expression of a receptor known as CLEC2. This is a C-type lectin receptor, okay, which is expressed to a much higher level in case of dendritic cell, in case when there is exposure to PDGF. Okay. This. So then we did another experiment where we mock transfected dendritic cells, no PDGF. We transfected the CLEC2 receptor because if CLEC2 is the one which is resulting in the induction of T regulatory cells, then you should not need PDGF there, right? So in this case, we just transfected the dendritic cells with the CLEC2, and this is the expression which is shown. And if you just take these CLEC2 expressing dendritic cells and put them with T cells, they are again making this high levels of IL-10, interferon gamma, at much higher levels of IL-10. And when you look for this, this is FOXP3 expression. This is much more, like 3.2% compared to 0.18%. This is more transfection. So you can repeat what we were seeing with PDGF with this receptor. So this tells you that this CLEC2 receptor is what is inducing your Tregs. But we really were not able to use this for therapy. Simple reason was the CLEC2, when we did a survey, it is also expressed on a number of endothelial cells of our vasculature. So you cannot use we wanted to use it for treatment in autoimmune diseases, like maybe an anti-CLEC2 antibody or something like that, which activates the CLEC2 receptor and now induces Tregs. But the problem is that the CLEC2 receptor, as I said, is expressed also in the endothelial cells and some other cells. So it would lead to some other problems in the vasculature. So we were not able to use this particular receptor as, or this particular finding as any kind of therapy. But we are still looking at other kind of, we have some other things like we use retinoic acid, which is vitamin A. It is known that it is present in high levels in the mucosa, and it kind of suppresses the system. Many of the tumors also produce. So now that knowledge, we are trying to see how it induces T regulatory cells, what kind of mechanism it is using. Because if you can identify that, you can use that to in autoimmune diseases where it is more overactivated. So knowledge from cancer, I know cancer has a number of things which is suppressing the immune system, but you can use that knowledge to apply it to something where you need to suppress the immune system. There is not one culprit because it depends on the antigen, self-antigen, which has resulted in that particular autoimmune diseases. Is it activating? What kind of a receptor it is activating?
Globulin against antigen, our own antigen, or because of infiltrations of T cells into the.
So it depends on the disease which autoimmune disease you are talking about. The major thing which is happening is that your immune response, your immune system is making a response against your self antigen. So the activation can be at the innate immune level first, which ultimately shows up as the antibody and your T cell filtration. So now the question is, do you want to act?
So we can interfere in any stage.
Any stage. That is the thing. Yes. So, and you also have to decide where you want to target your therapy. Do you want to make T regulatory cells in the end so that suppression is there? You can target, you can also, then you can do it in two ways. Either you target the innate immune cell to make the T regulatory cell, or you can infuse T regulatory cell directly into, you can generate them via some other means and you infuse them back into the body.
T-reg cells.
Yeah. There is a major focus on that also. A lot of people are working on how to get maximum amount of Tregs, maybe ex vivo, as what you were mentioning, outside of the body. Then maybe try to apply that in, inside.
But again, ma'am, lifespan is very less. No, only few weeks it will be there. After that, again we have to administer.
That is true. That is true. That's why, see, now there are two views for it. if you look from a pharmaceutical company point of view, they are
They want money.
They don't care if you have to do many times, because it's more profitable in that way, right? So that's why you will see according to me, that's why I see that most of the drugs which are for many diseases are more for maintenance therapy than for curing.
Yeah, they will not work for cure.
Yeah, most of the drugs are for, okay, they'll mitigate and then you should keep on taking them. Because if it is a cure, then it's of non use. Huh? Yeah, it's a one-time thing, right? So
Conditions
in the
in oral conditions, in oral autoimmune conditions.
I have only seen so far retinoic acid only in topical. So oral is not topical as such, no? Like it's going to go inside the mouth, right?
Yes, but there are a lot of medications which are applied orally.
Applied orally. So I have not seen any such use as such. So, but retinoic acid, what it will do is it's going to suppress the immune system. So unless you are using it for as an anti-inflammatory therapy, then it is fine. But for cancer, it will not be effective, because it depends on what you want to use it.
No, like diseases like pemphigus and pemphigoid, which are very painful.
I know. I haven't seen any use of it in that. So I'm not 100% sure that it will work, because I haven't seen any. Because in pemphigus, I think it's more the epithelial cell integrity which is compromised to a certain extent. The surface doesn't work that well. Yeah. So epithelial cells do produce retinoic acid, but I'm not sure that only retinoic acid will be sufficient to work as a cure or help mitigate the problem. But it may. I'm wondering if nobody has thought of it, and if it is worth thinking about in some way or the other to be tried.
Because we get to see a lot of disquamative lesions. So since you were talking, it suddenly hit me that it can be a good, sample size can be tried anyway just by topical application.
Yeah, because there's a lot of inflammation also in pemphigus, right?
Yeah. Yes.
So retinoic acid does work very well in inducing T regulatory. In fact, it is one of the mechanisms which is employed in our own body, in the mucosa especially. The epithelial cells produce retinoic acid, and they induce this T regulatory cells, especially in the intestine. It's been like shown very in more detail, because, and the reason I think we know that it is there in the intestine is more research in the mucosa has been done in the intestine than in other places.
So even if the person ingests, it's okay. Even if he swallows, it's okay.
It is a metabolite of vitamin A.
Yes.
So it should not be.
There are no side effects.
I don't know. So far the creams are all topical, right? On the skin.
Yes.
So ingestion I haven't seen. So, but if it is produced by the intestine, then theory it should not affect, right? It's going into the.
Theoretically, it should not.
Yes, but it's something which has to be tried then. So these two are just examples to show that how different compounds can induce different kind of TH responses, and how you can even use your natural compounds which you have, which you know have shown effect, and maybe if you can find out the active components of it, you can use them for as an immunomodulator. So this is another example of C-type lectin receptor DEC205. This is identified by Ralph Steinmein, who is the father of dendritic cells, and he has used this targeting to induce, again, Tregs. It induces tolerance in dendritic cells. So this is another receptor. So it seems to me that a lot of C-type lectin receptors, apart from dectin-1, seem to induce more like a tolerogenic immune response. CLEC2 was one. Then DC sign also some people have shown induces a little bit of tolerance. So apart from dectin, most of the other C-type lectin receptors seem to suppress the immune response. So this is, this is exactly what I had shown, that it leads to induction of FOXP3 cells, which will. So here he was using it for in the type 1 diabetes or type 2 diabetes, basically, where there's a destruction of type 1 diabetes, where there's a destruction of islet cells because CD4 T cells against islets are become activated and they're killing the islet cells which produce insulin. So now if you can induce tolerance, you have these FOXP3 cells which will suppress the CD4 cells, so no more killing of the islets. So you can use any model, whichever autoimmune disease model is in working in your lab. So a lot of people use multiple sclerosis, the EAE model, experimental autoimmune encephalitis. Rheumatoid arthritis is another one, and then type 1 diabetes is. I think these are kind of the major models which are used in most labs. The advantages of rheumatoid arthritis and multiple sclerosis is, in rheumatoid arthritis, it's easier to follow in such way that because there's a paralysis of the joint, so you can see that it is only in this area, or it increases. The mice is not walking this much, or this whole leg is not moving. So that way you can score the disease, how severe it is. Similarly, in multiple sclerosis, the same thing, that there's more paralysis, so the mice is not moving, so it's easier to score without killing the mice. So that's why those two models, I think, are used the most. So this is just to show that C-type lectin receptors are used in, they are very important for fungal recognition. And dectin-1, the scudlan which I mentioned is also a fungal molecule, right? Like a beta-glucan. So most of it is, we are reaching towards the end of it. So many of the drugs are now targeting this mannose receptor because it is a C-type lectin receptor. I think this has been the most targeted molecule in terms of C-type lectin receptor. So some people are even using this for CT analysis, this PET CT analysis. Because if you have nanobodies and things which can target certain receptors on cells, and then you do a CT scan, you can look at these cells because it has delivered the particular molecule which can be recognized by the CT machine. This is an example of the 99TC tectonium, or whatever it is known as. Okay, I think I'm more or less one or two things simply. So this is again inflammasomes. These are all the infections against which it protects us. And these are all the diseases where it is involved in. Just to show you that in diseases like autoimmune diseases also, now we are seeing this NLRP3 involvement because it also induces this interferon. So that's why it is being so these are various inhibitors which are being tested of inflammasome, NLRP3, Transilat, this. These are in various stages of trials because this is a very recent article, if you can see. So that's why I said these are active areas of investigation. But there are also this, so as I said, the IL-1. So basically the IL-1 is becoming unavailable. And then this is a decoy receptor which also binds this. And similarly, two other agents from GSK, Glaxo are coming, which are blocking the action of IL-1 or IL-18. So, and the reason there's so much interest is that these two antibodies have been shown to be effective in a number of diseases. It's not one. Even in diabetes, any disease you take, they're effective. That's why the companies are very much interested in developing these biologics against this. And metabolic related diseases are now becoming more and more prevalent. So I think this is probably the last of it. So I haven't mentioned here so much, but the toll-like receptor agonists, which the list I showed you, you can use them for vaccine adjuvants also, and a lot of them are, because so far the only adjuvant which have been licensed for human use in vaccines is alum. No other and alum has been used for since the first vaccine, I would say. After that, I think the new one is some ASO something which has reached 3 or 4 clinic, level phase 4 or something like that. So many of these TLR agonists, like they've identified monophospholipid A, muramyl dipeptide, components of LPS change to certain extent, so much of an extent that it doesn't induce any high level of inflammatory or other problems, because you see LPS was involved in sepsis also. So now so much of chemical structures and different combinations have been tried that now I think some of it have moved forward as adjuvants or even for treatment, immunomodulators, and not so much in cancer, mostly as vaccine adjuvants is what I would say. So that's also another area where these PRR agonists, antagonists can be used. Okay, I think that's it. So now if you have any questions, I can answer.
Ma'am, one question related to the rheumatoid arthritis. Actually, in case of the pathogenesis of the rheumatoid arthritis, the RA factor initiators are till not recognized prominently, because there are the RA1 and RA2 factors are there, and several one are going on and finding. But actually, who is the immunogens reactant for the causing the initiation of the RA factors?
That is still, so RA factors, some people think they're also acting as, they're also being recognized by some toll-like receptors, but it's more the joint damage associated patterns like synovial fluid or this hyaluronate and other things which are produced that seem to be activating more the immune system, at least the toll-like receptors and other receptors. RA factor, by itself, I'm not sure that it activates the immune system. It is one of the markers which gets upregulated, maybe as a consequence of immune activation, but I don't know if it by itself activates immune system or not.
The doubt is regarding with this because of rheumatoid. In case of the gout, there is inflammation in a certain part of the area, but in case of the rheumatoid arthritis, the immigration of the inflammation in the various part of the body at various times.
Yes.
According to the change of the climates, there may be a chances of inflammation in a various part, not the particular which will occur previous one.
Yeah, it can happen in different joints, right?
Yeah. So basically, is it any ideology that previous injuries sometimes occurred that will be lead for the initiation of the RA factors or the immunogens? Is
So-
maybe the reason?
So many times people believe that the initiation of an autoimmune disease, especially like rheumatoid arthritis or any other, it could be a genetic factor, but it could also be because of any kind of infections, as you are mentioning, or some kind of an injury, which really leads to release of factors which are which leads to overactivation of the immune response. Either some kind of a mimic comes in, or some danger associated molecules are released because of this, as you said, some injury. So it can, there is no one particular thing which can, which is considered the culprit. It is a combination of all three. Even in people who are genetically susceptible, you have twins and one becomes susceptible, like one develops the disease and one doesn't. So it is sometimes, it is believed that the combination of maybe environmental factors, which could be infections or injury, along with genetic susceptibility, play a role in this autoimmune diseases. Usually in autoimmune diseases, many a times you will see some kind of a family history. right? Maybe not, some are not transmitted like every generation. It could be some generations skipping, but it is still there. So that is all I can say at this point. Okay. Thank you. So tomorrow we will talk more about adaptive immunity, and then how you identify adaptive immunity related diseases. But we will talk about immunoassays from that point of view. So that way you will learn about the various immunoassays which are used for diagnosis and other things. I have to ask. Little earlier because today was the first day, so he wanted to make sure that all of you have time to come in and register and everything.
Tomorrow 10 o'clock. We'll start lecture at 10. We'll have breakfast and then we'll. Tomorrow 10 o'clock. We'll have breakfast here and then at 10:30 we'll start. Okay. If you want that we should start early, we will start early. $10. Fine? Okay. Do you have any suggestion for me? Any feedback? No? Yes, ma'am.
First of all, thank you, sir, for giving me opportunity. Actually, I'm your student, so no suggestions, but the madam was very good in immunology, and I have not being taught immunology since 2.5 years. So it is very interactive session, as well as it is very basic for me, as well as I have recalled all the basics of immunology as well as the research, and I could find the connections because uh, recently I have written one chapter regarding the cancer and as well as the targeting of this particular cells by the uh, hybrid nanostructures. So I have gone through these things, so it is little bit connecting. So
Thank you. Okay. Fine. So we'll stop here today. We'll meet tomorrow 10 o'clock. Thank you.